tell you what is ImmunoCure, okay, to have a small what we do it, okay. So we are actually a small molecule drug discovery company, and also we do some, vaccine modeling, other things for our clients based on our expertise. But our company mainly uses, we can call it as a data-driven company, where we use our computational and also artificial intelligence methods, which was served with respect with our in-house databases. Based on that, we generate the models and predictions, and using that, we design and predict the new molecules. And then we do the synthesis in our lab and do the testing. So everything we do it, and then we provide it to the client. Okay, this is what the molecule you have, new molecule, and this is the activity. So we do complete POC for the client. But it is not something like a traditional way. We use all novel technologies, especially cutting-edge technologies like AI and computational methods, to come up with a new molecule where we need not to synthesize huge number of compounds. We can use synthesize very less, and then we can get the actual activity what we are looking for it, and also other multi-parameter optimizations. Okay, our main in India. Main office in India, especially in Genome Valley, near in Shamirpet. Okay, and now we have an office. Recently, some part of our company was took over by the US part, ImmunoCure LLC, and it is in Brooklyn, New York, as such, right? And if you want to contact, we have ImmunoCure US and ImmunoCure. And a small introduction of our team: me, Nagesh, and Kranti, Dr. Sumana, Linga Reddy, and Prashant. These are the key people who are the pillars of the company in various models. And then, yeah, we have
Repurposing Polypharmacology.
All right. Yeah. So now we have AI team where they try to generate the models, and then they will provide us to our computational and also for our chemistry people, bench chemists, to which molecule to be synthesized and everything. Okay. And yeah, so we finally do with our tools and apps, whatever we have it. Target validations, hit to lead and lead optimization, and in the discovery chemistry and the biology. These are the things which we cater to the client. And we can say the data-driven method, but I don't want to put much in that. And we use all computational capabilities, whatever in lead identification, optimization, and also for the ADMT modeling. And one of the very important backbone for the company is we have a very good collection of data. This is very important even when you do the repurposing. Okay, so we have a very good collection of databases in various stages of the drug discovery. These databases are nothing but which was experimentally conducted and published in various literature, like journals and patents, and also from the commercial sources. Okay, so we collected and curated, and now those databases we rigorously use for model generation by using the machine learning or computational methods, and then we use those models for predicting the new molecules, maybe for the can be used for the repurposing message. Okay, so and we have the chemistry services and so good a beautiful lab for the chemistry analytical, and also have a biology services. Okay, so something like ImmunoCure discovery, when you put together in one slide, this is what we do it with all our cutting edge technologies as such. Okay, ImmunoCure proprietary database with a druggable and scaffold library, hit to lead. All these things we use our computational and machine learning methods, and then we do the synthesis and bioassay. So this is what we do it, and we generally collaborate with the clients, and we do something like a milestone based, like hit, lead, and lead optimization, or maybe something like a FTE based. That's what we do it. Okay, this is what about the company, and we also have internal research. where we, we came up with the molecules, especially in the oncology, or we can say the broad spectrum oncology, and also in the immuno-oncology. And in the immuno-oncology, we have a repurposed molecule. And that I cannot disclose all the things, but what is the workflow, what we did it, we just put it there. And also we are working in the antibacterials as an in-house discovery. So that's what our company does. Completely we do only research. That's what in the company. Okay, no CRO. Okay, now we come to our repurposing and something like workflows, I can put it, and case studies. So whenever we say repurposing, this is one of the very important things what we can understand or should be known. So we can do repurposing when you have a good database of a known history of compounds and their properties, maybe biological properties. Okay, when you have these things, then we can start with the repurposing of any molecule. We can connect them maybe by using what you can say, that artificial intelligence, or maybe with other ways. Okay, we can connect the dots of these molecules to the drugs, and then we can come up with we can do the repurposing. Okay, and one thing we should remember that, okay, we have our mainly body is made up of various proteins, and they have different architecture, etc. But still they have some similarity. One molecule can bind to many number of targets, which is called as polypharmacology now, and it may lead to the efficacy against the particular disease, and also results in your side effects or adverse effects. And sometimes we don't know, it may be useful for curing the other kind of diseases as such. Okay, as we know that, okay, after post-genomic sequence human genome project, there are many targets have been discovered, and we are still in the process of getting the druggable nature of each protein, such that which can be used for various diseases as such. So now when you say that, okay, whenever you say druggable target and biological targets, okay, so we need to remember that you should have a collection of, at least you need to have a collection of targets. What are the targets which are there, and what are the clinically trial in the targets, what are the patents targets, means how people are working, what can be a druggable target, simply we can say that, okay, and how many molecules which are there which are binding to those targets. If you have this collection of molecules and their targets, okay, and also provided with their adverse effects or any kind of toxicity issues for those targets, then this can be a very good starting point for the repurposing actually, or maybe in the drug discovery, it might be anything. Okay, so that's what any people, okay, even this is what I will say, that whenever you say the repurposing are data-driven methods, okay, James White-Black, who got Nobel Prize in 1988 for the development of propanol and cimetidine, okay, say the best way to discover a new drug is to start with an old one. Okay, so from the old one you can come up with a new drug, or from old one you can come up with a new therapeutic application. That's what the repurposing. That's two different things. Okay, so always, old understanding in a proper way, systematic way, is the way to come up with a new therapeutic of a drug, or maybe come up with a new molecule and see. So simply we say that therapeutic target, everybody knows that every therapeutic drug action generally occurs by changing the activity of the functional protein. We'll call it as a drug target, which maybe upregulates or downregulates the target activity. Based on that, okay, it can cure the disease. So sometimes in the disease condition, the target might be upregulated, which results in the disease. So our drug will downregulate that, and then reduces the disease condition. Or sometimes you want to upregulate, the target is not upregulated, you want to upregulate that. So we should have a drug to agonize to increase the activity of the target as such. Okay, so that's how this drug target is one of the vital, and which makes them which cures the disease because of its malfunctioning. Now, so if you put together this target, okay, so generally we can say that the targets might be a protein or DNA and RNA, okay. So mostly DNAs are mainly targeted, especially on the cancers, and also RNA. And moreover, in the viral or bacterial infections, okay, DNA and RNA are the most vital target for it. And therapeutic targets, if you see that, okay, if you put together all the targets, we have something like, druggable target, whatever I say that, which is there in the literature and the patents, etc. We can say that 3,419. Okay, all the targets we put together. Maybe I think that made this presentation maybe in just 6 months back, maybe now maybe 3,600 plus, something like that. Okay, so if you see that the targets, whatever we have, it is very less number. Okay, which is there in the successful target, means which are approved as a drugs. Something like is only 461, and a clinical trial targets is something like, it is 1191. Okay, clinical trial targets, and it is a little bigger number because a lot of molecules are there in the clinical with new targets, and patented targets are something like 201, and research targets are 1500. Okay, so something like all the targets are something like it is very less number, and these are the druggable targets which are there in the literature, and people are coming with a new target for new efficacy. And now when you see that all the drugs, this is something like which we say that, nutraceuticals, everything if you put together, all the drugs is satisfied, but the approved drugs, whatever small molecule approved drugs, will be around 2,649, or maybe you can say now another 100 we can add it. Okay, so around 2,700 compounds drugs are there, and we have around 10,000 clinical molecules, and patented something like, molecules are 5,000. It's not the drugs, it's a patented drug-like compounds, and experimental molecules, experimental drugs are around 20,000. So this is a number. So now, sorry, when you put together, okay, the approved drugs, clinical and patents, we got something like 37,000 compounds. Okay, so now we have 37 is a small number. You know that when you synthesize any molecule, the chemistry, if you put it, the chemistry of chemistry space, the total chemistry space which can be synthesizable, it is something like 60. 2 to the power of 60. That means a huge chemical space. In that we have only 37,000 compounds which we are playing. In that we have only 2,700 approved drugs, and others are in the clinical and other stages of it. Okay, and now we have 3,419 targets. Okay, and now many new targets are available. And now if you systematically understand these targets and the drugs, okay, how well they are doing it, okay, and put together in a form of acquirable database and make it as connections, then definitely these results in the repurposing, or maybe in the what you can say that new drug discovery as such. Okay, and there are several sites available, okay, in the internet, because internet is a very good wealth, I think, for these resources. Okay, one of the important website and the online database. Okay, always I will tell to the pharmacy and also the pharmacology people and the biology people, you need to always go through this site, guide to pharmacology.org. Okay, where they will provide the what you can say that all the guide based upon the International Unions of Basic and Clinical Pharmacology and Tissue Pharmacology Society. Okay, this was actually this was developed by the European Union. Okay, and we have the site guide to pharmacology, where it has the drugs and experimental drugs, clinical drugs, and their efficacy, and also their polypharmacology, and also therapeutic. What you can say, side effects and other things. Okay, well documented in this. Okay, so please go through it, and that will be a very good starting point for you people when you are working in the repurposing area. And also there is another tool, Campbell, or I think Campbell 2021, which is updated one, has a wealth of the molecules, whatever you find, all the 37,000 molecules. Along with that, any drugs in the literature, in the journals which have been published, okay, will be documented in the Campbell. Okay, so drugs, their therapeutic efficacy, and if it is having any off-target effects, if it is binding to any off-target and was reported, is documented in the Campbell. Okay, off-target is nothing but polypharmacology once again. Okay, so which is binding to other than the primary target, we'll call it the off-target, which is in the sometimes useful or maybe in the mostly in the side effects as such. And drug central, cancer, and drug data bank. I didn't put that, drug data bank. Okay, so these are the sites where you have these all these data available with different targets, and also properly categorized targets, and also the the therapeutic efficacy. Okay, now we have, because what we were doing, just simple thing, any company, if you do this kind of proper systematic understanding, even you can start a company and then serve the clients as such. Okay, that's a simple way to do that. Okay, so you need not invest much about money to start all these things. Okay, but you need to understand very well about the database, some concepts of the statistics and the computational things, so that you can properly organize it, and then you can start serving to the clients. Okay. Yeah, so immunocure, actually, we have all these data and properly curated, including the experimental and patent, and also the other, which was not, available in this, okay, and which was reported in the journals and in the commercial libraries, etc. We have made our own database, and then this pool of database we're using in our in-house discovery and for serving the client. And generally, we don't provide the computational machine learning methods to the client. We try to use internally for the client drug discovery. Okay, so we never do the computational or AI services to the client. Okay, we use that for our internal discovery of it as such, right? And now, when it comes to the target, little bit about the targets. Okay, people might be, most of the companies, what I saw, a few companies in India, when I contacted them, how they are doing it. Okay, so what they do is they just simply collect the data from various sources. Okay, and then they will try to generate some models, and then they try to predict the models. Okay, they try to generate, because now artificial intelligence and machine learning tools, okay, whatever, maybe in the previous lecture I told that, okay, these methods will be useful to generate the new chemical space. Actually, this is called generative chemistry, where it try to generate a lot of combination of molecules, okay, by using these artificial intelligence tools. So now these molecules can be predicted by using the known databases as such. Okay, now when I approach one or two clients, okay, two artificial intelligence companies, okay, that how well they are working on it, okay, to understand them as such. Okay, so what I understood is those people, they are good in competition, in programming, and they are very good in in making the visualization and everything. But what the problem is, they are lacking the biology aspect. Okay, this is what I understood, that because now the most buzzing thing in the industry is AI and also data science. So most of the computer scientists, okay, they are just doing these things, and just generating some models based on the data, categorize it, and then generate the models and predict it, and then generate the new molecules. All these things are automated, and then they give the molecules. Okay, but what is the problem here is drug discovery is not the repurposing, whatever you say, is not that simple thing where if you do some automations, okay, cannot give a molecule as such. Okay, so it cannot be something like from computer can generate a molecule. Okay, so we need to understand the biology. And God is not made as a very simple, they made it more complex actually. Okay, So they categorize simply, if you put our body, we have a proper categories of targets, okay, something like GPCRs, okay, all the targets, whatever we discussed previously, may be categorized into GPCRs, okay, as a based on their function, or based upon their, what you can say that, architecture, okay, ligand gated ion channels, okay, and transporters, voltage gated channels, nuclear hormone receptors, kinases, okay. So like that, we have having based on their function or their topology or based upon their activity, we have different class of targets, and they share some commonality because they do the similar kind of function. Okay, and when you go to that, okay, when you go to each target, okay, once again, they have a main target, the catalytic site, allosteric site, okay, they work as the agonist and antagonist, something like that, okay. So each target and each class of target, it has a lot of complexities, okay. So sometimes the protein is inactive state and active state, some molecules bind in the inactive form and some molecules bind to the active form, and so it's all, I know it's a when individual nature of each protein, how makes a function of the protein. So now when you understand little more about each target, okay, and how it functions, and then based on that, if you generate our models, okay, and that will be more precise when compared to large scale categorization and providing the solutions. Okay, that's what I just found with these companies. They are lacking with the biology understanding, how these targets and what is their nature, and how ligands bind. So which molecules bind allosterically and how which molecules bind catalytically, and in inactive mode how they bind, intermediate mode how they bind. So if we have a good understanding of all those things for each targets, okay, and then if you make the models, then our models will be more precise in finding the new molecules. Okay, yeah. So, and now when it comes to these GPCRs, okay, now when it comes to the drugs, whatever we have it, okay, and also the available in the Campbell molecules, most of the molecules, okay, we can say 50 to 30%. You can say, if you see, the A is drug molecules, okay, so 56% of the drug molecules are from the GPCRs, so approved drug molecules.
నేను యాక్చువల్గా స్పాట్ గురించి కాదు, ఇది ఆర్టికల్ చేస్తే
and so 19% from ion channels and 10% kinases, etc. If you see this, many drugs which are there in the market, okay, we can say 2000 3000 compound, in that around 56%, maybe you can say that 1400 molecules are against GPCRs. And GPCRs are 7 transmembrane proteins, and it has a different categories, okay. Like GPCR class 1, R1, etc. They have different categories, but they are the transmembrane proteins. They have some commonality. Okay. In the same way, if you take the kinases, okay, kinases are the proteins where they have some commonality, where the ATP will bind to the catalytic site, and then it will do the phosphorylation for the energy of a cell. So that means there are, if you say that there are around 250 kinases are there in the human body. In that 250 kinases, okay, all the kinases work as the ATP, phosphorylation. And if you find, if any molecule is binding to, if a molecule is binding to one kinase, it can bind to other kinase, okay, because they have a common catalytic site, okay. And even the GPCRs, ion channels, etc. Okay, nuclear hormone receptor. That means that if one molecule is binding to one target, okay, it can bind to other target because there are some similarities between the process similarity between the other proteins. Okay, now if you crack the proteins which have that similarity, okay, between the proteins, and then if one drug is binding to a particular protein, if it is binding to other protein which has some therapeutic efficacy, then you are going to have a repurposed molecules. Okay, so this can be happened when you have systematic understanding the, what are the targets and how they are function, and what are their, inherent properties of each target and how they are binding, and what is the topology of the molecule and what is the topology of your target at the binding site of where it is binding, or something like, we can say that what kind of amino acids, what kind of pocket. If you have understanding and the correlation between them, then we can come up with a new molecule for it as such. Okay, or you can repurpose the target. Okay, so targets, we need to understand them, and then we can see how these molecules are binding and what are these molecules. Okay, what is their topology? If you understand that, okay, then we can understand that how GPCR pharmacophore, okay, helps to, uh, to bind to a GPCR molecule. Means GPCR molecules, which pharmacophore makes it bind to the GPCR, or maybe kinase or something like that. Okay, so now we have this, as I told you, we have a repository and we categorized these databases, okay, along with the chemical and also the biological function, and we correlated them. Okay, and we saw that what is the cross interaction between this, pharmacology and the molecule, and what topology makes the polypharmacology be correlated with that as such. Okay. So this is what about something, whenever we see the drug targets and the molecules, so we need to remember that we need to understand the targets, okay, when you go for, before going for the repurposing of it, okay. And now, simply, if you want to say that the discovery pipeline, so we can say the target identification, validation, lead optimization, preclinical, I just forgot to put it, yeah, and then clinical trials. So we use a lot of bioinformatics, structural informatics, and then we'll come up with a drug discovery. But when it comes to the repurposing, which we call it as a polypharmacology, identifying a new therapeutic efficacy, okay, the people are very crazy about it, and especially formulation industry is very crazy about it, because the drug discovery itself is a big, money churning and time consuming. So we need to invest, we need to pour $3 billion to get a new molecule, to new drug, from the bench to the clinic. I mean, into the not to the clinic, a bench to the drug store. So 3,000 billion in any step, if you fail, then you are going to put the money in the vein as such. Okay, so that is a big challenge in the drug discovery, and it has a lot of attrition. And also it takes a lot, 12 to 15 years. So you need to put money and also time. And finally, we are in finger crossed whether we can reach that goal or not. But once you get a molecule, then it is $1 billion as such, multi-billion dollar. Okay, that's why even though it is a money, investing thing, but the pharma industry, pharmaceutical companies put money on the research to come up with the new molecules into the new therapeutic area. That's how research grows, and also, once you get a first-in-class molecule, they earn a lot of money from out of it. But for the existing drugs, okay, if you find out a new therapeutic efficacy, we'll call it as a repurposing drug, repositioning, a reprofiling, a retasking. Okay, something like a strategy for identifying new indication for an approved or investigational drug that has not been approved. So either for an investigational or something like in the clinical trial, some molecules might be discontinued for some other reasons. Okay, if you identify the new therapeutic efficacy for it, so then we can say, it's a repurposing. One of the very, 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 classical example what we can give is a thalidomide. Okay, this is mainly now repurposed for, especially for the anti-cancer function, especially multiple myeloma, sildenafil, and erectile dysfunction. Remdesivir, very recent molecules, which was, used for the COVID treatment, but we have, different, opinion about the remdesivir, but how well it is effective with the COVID. But yeah, but in cellular level, okay, they are very, very effective, remdesivir. Minoxidil, which was mainly for an antihypertensive drug, which was now, I mean, sorry, for actually, it's not R, F for, it's a type of mistake. Especially now they are used for the hair loss, and that Redis has one of the formulation for the minoxidil for the hair loss. Okay, so which was actually for hypertension. Now way back in 1950s or 60s they found this minoxidil. Now they repurpose for hair loss as such. Okay, so there are repurposed molecules available in the market for the different therapeutic function, and now we have drugs available in the market, and also new drugs are adding, new targets have been discovered, new pathways have been discovered. Okay, and now we can repurpose our existing drugs. Okay, now we can reduce the money whatever investing for the new drug discovery. But only thing is the major challenge, okay, pitfall of the repurposing here is maybe we are finding, digging our own mine, so that we cannot come up with a new molecule which can be used for the, the new therapeutic area, new functions as such. And not only that, as this molecule is repurposed, actually it is meant for one function. If you are looking for the other function, sometimes, it may not work for large population or something like that. Okay, and also, the efficacy is a little bit in stake. Okay, so if you see that, remdesivir, that is what, in some cases it works, in some cases it will not work. Okay, but yeah, so, and because the safety of these drugs are, already been tested in the clinical trials, because these are all drugs, repurposing will be always rapid and cost effective. Okay, that's the reason. If you come up with a new drug, okay, based on the target and also the the different platforms or AI methods, okay, if you come up with a molecule, okay, and then you can license them, okay, and then once you get the therapeutic efficacy in the preclinical, especially in the animals, or maybe in the phase 1 clinical in the animal studies, okay, then definitely immediately this molecule need not to face the phase 1 clinical trials. You can directly go to the phase 2 and then phase 3 clinical trials. Especially they look for the efficacy, how well it is reducing in the human. Actually, they because already the safety have been established, and now in the phase 2 and especially in the phase 3, they see the broad spectrum of safety, but mainly focus on the efficacy of that molecule as such. Okay, that's how we can reduce the repurposing something like from 15 years to 3 years, minimum 3 years. Sometimes, especially some kind of molecules in the rare diseases, etc., you are repurposing a anti-cancer compound from some rare disease or some other disease. So we need to go for little more stringent rules, because so anti-cancer compounds are more toxic. Okay, still they approve it because of life-saving thing. So that's the reason. Okay, if you repurpose any molecule which has some toxicity, still got approved, then they have some more stringent process to be done. Okay, otherwise, most of the times, 3 to 5 years, repurposing molecule goes into the market as such, right? And there are many drugs, okay, which are there, and some of the drugs which was approved by the Food and Drug Administration. Okay, and different orphan diseases and also the common diseases. Most of the times, okay, these drugs were intended for the orphan disease, because whenever you see the repurpose, okay, we can see the orphan diseases and also the rare diseases, actually, because most of the times our big pharma industry neglect these rare or the orphan diseases where they don't have much especially rare diseases, they don't have much commercial output. So people will repurpose for this and fund for this, rare diseases as such. Okay, for the repurposing of the molecules. Okay, so now we'll come to a little more into that. Okay, repurposing, how we do that. Okay, so identifying the polypharmacology for the drugs. Okay, now we have a drug molecule. Now we have primary targets, and we have off targets. So when you put together, and see how they are binding to different targets, or maybe in the pathway, how they are binding. If you understand these things, okay, then we can do the repurpose, basically by systematically studying their disease commonalities, okay, and their pathway of these drugs, how they are binding to that, and how the targets are similar to each other, okay, where where the drugs are binding, and bound ligand similarities, something like, similar binds to similar. If you have similar ligands, they can bind to the similar targets, okay. So that's how bound ligand similarity, drug disease similarities, okay. So based on all these things, okay, we can do the repurposing and come up with a new target, okay. Something if you put together, okay, computational approaches, okay. Okay, repurposing is one of the very good example. Our computational tools are very good examples, especially for the repurposing of the new molecules, especially A and other computational chemistry methods, okay. And there are a lot of, most of the remdesivir and different maps against the COVID was repurposed by using the computational methods as such. Right? Now, when you put together the important computational methods, okay, especially for the repurposing here, okay. So chemical similarity, comparing the something like chemical similarity. If, as I told you, similar ligands bind to similar targets, okay. So now, when you have different molecules, okay, different molecules which are binding to the different targets. Now, if you cluster these molecules, if you find similar molecules, okay, most of the similar molecules will bind to the similar kind of targets or having some similar topology in the target as such, okay. So now if you understand the drugs and their chemical natures, okay, and then compare with the label, because when you go to the literature and PubChem and Campbell, etc., you can find a lot of molecules which was categorized for different functions and different therapeutic effects and different targets, etc. Now, if you take a drug and compare with the PubChem database, or maybe the database with the known targets, okay, then if you find any molecule which is very close to your drug molecule, okay, then you can see that the new molecule, the molecule which was labeled or which was already known which target it is binding, and that is similar to the new the drug, whatever you are looking for it, then you can say this drug can bind to the particular target, and then I can start working on it. Such, okay. So chemical similarity and target similarity, okay. So another thing is, if this molecule is binding to a particular target, and this molecule can bind to other target which has shared some similarity. If the new target, if it is binding, okay, if this new target has having some different function or different therapeutic efficacy, then definitely this drug can cure new diseases on the new target as such, okay. Target-based similarity. People try to use structure and sequence similarity, or network. They make it as a network, ligand similarity and the structure similarity, and then finally come up with a new molecule against a new target as such, okay. They use computational methods like molecular docking, or maybe a automated a engine for the docking to come up with the target similarities as such. And pathway analysis, drug disease pathway analysis, okay. People look into that, and then they try to see which pathways these drugs are binding, and now in that pathway we can come up with a new target where it can have a new therapeutic efficacy, okay. And estimating the drug disease similarities, okay, we call it as a transcriptome analysis, okay. So in all these cases, okay, in the computational methods, okay, when you want to do this study, remember that we need to have our own known database, a collection of known database. As I told you previously, where I can get this database from: Campbell, PubChem, maybe Guide to Pharmacology, Drug Bank, etc. They have a proper categories of the molecule drugs and the known targets and the molecules which can bind to the known targets, and their some other proteins like, databases like PDB, which will give the target topology and the three-dimensional structure, and the sequence similarity like the databases like Uniprot, etc. So these are all provides the information of the databases. Now we use these computational or machine learning methods to compare one molecule to the other, or to compare the molecule to the target, or compare molecule to the pathways, or maybe the drug to the disease. All these things we need to do it, connect them and make a model. And now we put in our drug, whatever we want to investigate for the new therapeutic efficacy, and put it into this model and come up with a new therapeutic index. Okay, right. So what is the main base for it is the databases and categories. Now we do the similarities and chemical similarity, target similar, pathway similarity, and everything to that as such. Okay. So in more, what I say that drug, target, disease, these are the three things. Okay, in the repurposing. Okay, we need to have our drug molecule, what will be the target and new target, and this target where, what kind of disease it can cure. Okay, this is what. So we need to connect these things. Okay, to connect these things, okay, we have different methods like, especially, the drug and their data and side effect data, whatever you put together, it's a huge data. And the ligands and the proteins and the targets, if we put together, we have almost 30,000 targets. We have it as such known targets. If we say that around 4,000 to 5,000 targets we have. There are many molecules which are, we have it. It's a big database. We have it with the targets, known molecules against different targets. Okay, and now we know that these targets in which disease it is binding. This is what we know it. Now we use this whenever you have a huge data, we use some computational methods for this data mining, especially. One of such is, machine learning with respect to this ligand similarity. One of the things, as I told you, is chemical similarity. So we use rigorously these machine learning methods, and then we identify the pattern of the ligands which will bind to a particular targets. Okay, and then particular targets. Okay, and cure the particular disease. Okay, or we can say that we try to identify the patterns of the drug molecules which can cure a particular disease. Okay, particular disease. Now, how I can do this? We have from the PubChem or from the Campbell, whatever the drug bank. Okay, we try to take those molecules, and then we try to see on particular disease or cancer. Okay, I'm working on it, and especially on the small cell lung cancer. I'll take all the known compounds from the literature or from the patents, etc., and see what is the pattern of their, are their pharmacophore, are their patterns, are the substructure patterns, are topology, are physiochemical properties, okay, which makes that molecule bind to have bind to a particular target and reduce the small cell lung cancer. Okay, now if you have that model with you, okay, if you generate that model, now if you apply the same model to a drug molecule, now if you find a new molecule, maybe anti-diabetic or some other molecule, okay, which have the similar pattern, then definitely this, if that new molecule, new drug which is there in the drugstore, if it is changing the similar pattern which can cure the small cell lung cancer, then now the anti-diabetic drug become a anti-cancer molecule. Okay, one of the example, glipizide. Okay, so if you go back, okay, you just see the polypharmacology of the glipizide. Okay, they use glipizide for the anti-cancer treatment, and also in combination with the other anti-cancer drugs. Okay, they use this glipizide for cancer treatment. So it's one of the repurposed molecule which was done in the similar purpose and such. Okay, so now what is the thing is, now we use the statistical pattern recognition for a particular disease or for a particular target, and that we implement for the known drugs and see any pattern is matching to that, and then we can come to up with the new therapeutic efficacy. Next, data mining and the data exploration and data analysis and visualization. Okay, now we do that. Okay, a lot of something like, we say that, text mining and find it out. Okay, if this drug is binding anywhere in this particular target, if this molecule is having any off-target effects, and if these off-targets are having some therapeutic efficacy, if that is been mentioned anywhere in the literature or in the net or maybe anywhere. Okay, we do a lot of data exploration, text mining, and data analysis, and come together and visualize this and connect to the nodes. Okay, and say that, this drug, okay, has this kind of side effects, and this side effect, okay, because of this target it is binding, and this target is having this kind of function, and this function will useful for curing this disease. Okay, if you connect all these things, okay, then we can come up with a new repurposed molecule. Okay, so this data mining, in a proper order way, and finally visualizing them in some statistical way, we can say that, we can repurpose the molecule as such. Okay, and statistical machine learning and data mining, okay, they are, I just put it in a different way, but they are very close to each other. Okay, sometimes they share the commonality of it. Okay, and then network analysis, actually. Okay, so now what we see that biological systems. Okay, now you will see that, what are the targets it is by the drugs are binding, and what are the if that's if the another target is binding, if the two drugs which are binding and they have a two biological networks. Okay, and they share some commonality between them. Okay, and then say that, these two drugs, okay, is sharing some commonality. Okay, and the similar class of molecules which are sharing that can bind to the target. That's how we can do the network analysis and try to connect the missing links for it as such, right? And also transcriptome signature. Okay, that, gene expression data, if you see that and see how well it is expressing the side effects, upregulation and downregulations. Okay, and then our drug gene expression data, how it upregulates and downregulates, if there is an opposite action, comparison. Okay, if you see that if a drug is upregulating, okay, and their particular properties, okay, and that is downregulated in the disease, that indicates that, this drug can be, used for the particular disease as such. Okay, and a protein comparison, this is something like, it's a complete bioinformatic analysis where we try to see the drug which is binding a particular target or to the off targets. Now, if any more proteins are there, or in any druggable targets are there, okay, in the human body, which share the same commonality and have different therapeutic efficacy. We try to find that, and then definitely if you find any new protein which share the commonality with respect to the primary particular drug target, then this drug can bind to the new target, and then this new target can use for different purpose as such. Okay, and in this, okay, another way people use it is maybe little more crude and time-consuming methods, which are called, but very precise, molecular docking and dynamics. They try to take all the targets of different proteins and for different therapeutic purpose, and try to take each drug molecule and see how well they are binding to each target, and see how well they are binding, and if it's having that pharmacophore with that, and how well in dynamically how well they are stabilizing the protein. They try to do that by using the molecular docking and dynamics, and then they found that this molecule is locking very well inside the pocket of the receptor. Then we got a new lock for the old key, something like that. Okay, so the drug repurposing we can put together. We need to find a new lock for a old key. New lock means it's a new target and different therapeutic purpose. For that, we took an old queue and finding a new lock. Okay, so randomly you can put in each in and then try to unlock it, or some rationally by using machine learning, network analysis, transcriptome comparison. Okay, we try to find out the locks which are very similar to each other. Okay, based upon their pocket as such, and now we try to put our lock into that, put our key into that, and try to unlock it as such. Okay, those are all different ways we do it. Okay, but identifying the new lock therapeutic lock for a old key. That is what the drug repurposing in this. Okay, something like chemical similarity. This is what I just put it because people will understand very well. Okay, chemical similarity methods. Okay, what the people will do and compare it as such. Okay, structure and chemical properties of a molecules are responsible for the therapeutic effect. Everybody agrees, no? The structure and the important features, we call it as pharmacophore, and which is complementary to the particular target, and then it will bind tightly and then do the, up regulation or down regulation of that protein, or inhibit or activate that protein as such. Okay, so we need to have some common, they should have functional group. Okay, so if you have a molecule very close to that, okay, this is a molecule, for example, and this is not a drug. It's a chemical molecule which was identified. It's not a drug molecule. If you identify from the drug, these are all the 2 to 5 drugs, for example. Okay, here. Now, if you identify any drug which is close to this molecule, and this molecule, first molecule is binding to some GPCR protein against some diabetes. Okay. Now, if you identify any molecule from the drugs which are very close to that, then these drugs may not be for the GPCR, or maybe they are for different, maybe anti-cancer compounds. Okay, others are anti-cancer compounds. If you find any drug which is very close to the GPCR binding anti-diabetic molecule, then this new drug anti-cancer compound can become an anti-diabetic. Okay, or vice versa. Okay, something like, if you find it, means if you have a chemical similarity between the molecules, then because of the sharing the chemical similarity, then the molecule which was designed for some other purpose can bind to the other target because they are sharing the chemical similarity, because the protein also may have chemical similarity, structural similarity as such. Okay, so now in this repurposing, explore the repurposing opportunity, repositioning up for a drug compound based upon the shared chemical similarities. Okay, so people generally use the clustering methods, or the constructing a network, okay, based on the chemical similarity, and identify the drug to the, chemical similarity between the drug to the targets. Okay, and they will connect it and then try to identify the new molecules as such. Okay, and just I will give a small case study here in our company, what we do with the kinases, chemical similarity, right? And so this is one thing I just always come across, people, some computer scientists who are listening to that. Okay, so people generally use smiles to understand the structure, okay, and compare the structure, okay, based on the similarity. Okay, and smiles is a notation, it's a 1D notation which represent a molecule. A part of a smile will be a fragment present in that molecule as such. You can see that the colors are nothing but the patterns of it. Okay, now when you see this, we can see the green color pattern, okay, might be important for the anti-cancer, for example. Okay, and now from the known compounds, from the PubChem or somewhere, or Campbell or drug bank, I got from the anti-cancer compounds, I know this is the pattern, the green color is the pattern which is for the anti-cancer. Okay, now if I take this pattern and search in the database of the drug database, okay, now if I identify any new molecule and non-anti-cancer compound, okay, a new molecule, non-anti-cancerous compound, which has the same pattern, then definitely that can be anti-cancer. Okay, that's how we can do the repurposing. Sometimes people use the fingerprints. Fingerprints will provide you that the future present in the future. If the future is present, we'll call it as an on bit. If the future is not present, for example, if it is the aromatic is not there, something that it says 0. Okay, so if feature is there, then it's a 1. If the feature is not there, it says 0. Okay, based on that, okay, fingerprint, okay, we can understand the topology and the functional groups present in the molecule. And the fingerprint will be very unique for each molecule. So based on the fingerprints or patterns, okay, we can compare one molecule to other molecule. Or we can say which molecule is similar and which molecule is dissimilar. Okay, in the same way, even in the protein also, we can compare based on the sequence, okay, how much they are similar to each other. Okay, structural topology. Okay, So now the thing is now, okay, that's good. Okay, so when you have this type of methods and data mining methods, etc., available, okay, now what is a major problem is, okay, previously we have very small systems to process a large data, very big data, okay, and it's very difficult to do the calculations in the smile side. We need to have a proper computer power, and also the sophisticated statistical methods to compare the data. Okay, now in recent times there is a lot of development in the especially machine learning algorithms and their automations, okay, which is called as now AI. And also there is a very big development, a big change in the in the machines, okay, especially now previously we have CPUs, now maybe cluster and all those things are there. Now it converted into GPUs. In a small machine we can have a GPU card and we can run these calculations, okay, and these GPUs are well used in that. And now recently, which mainly for the machine learning and AI architecture, we'll call that tensor processing units, okay, so which is been started with the Google TPUs as such, okay, that is a motivation where it has a proper network for these GPUs. And these GPUs when come into the picture now, the new sophisticated methods will call it as a deep learning methods, okay, which needs high computational power, okay, and to screen large databases, okay, okay. Now these GPUs are helping us to do that as such, okay, and reasonably we can do with the small databases like Bayesian and other Marco or something like other statistical machine learning methods, okay, especially when this GPS came into picture. Deep learning methods are now very much promising and doing a lot of applications, okay, and not only in the drug discovery, in other areas as such, okay. Now quantum computing is another thing where it is going to do another big noise, okay, in the coming years, okay, especially qubits, which were much more processing and can be, the new era of it actually. We can say now, previously quantum mechanical calculation, especially in the computational chemistry, we mainly rely on the molecular mechanical calculations, which are not accurate. They are approximate and relate to as such. But quantum mechanical calculations are very accurate, and which cannot be implemented, especially for the proteins or biomolecules, etc., because of the the computer power. Now QM computers are helpful in doing these quantum mechanical calculations even for the proteins, etc. Okay, that's how we can increase the accuracy. Maybe in future, okay, we can make much more better predictions, or we can design some molecules which can directly from the computers as such. Maybe can happen actually, maybe in the future, okay. So right, now this AI power increases and you, sorry, yeah. So how much time I have? Something
Yes, sir.
So,
Can
okay.
you please shut off?
Right. Sorry, I missed. Okay. So I just.
Thank you, sir. Sorry.
Right. Okay. So I just, go through the different algorithms. Okay, I should skip it. Okay. Right. Okay. Okay, so one of the very important thing which was machine learning started and useful and showing the mark, okay, especially with respect to the oral antifungal drug, which was in 1996. Okay, people use it for especially fungal treatment, skin treatment. Okay, but because of the toxicity, etc., there are few deaths happened, okay, because of TBFA, which is a metabolite which will do that, liver toxicity. So that was discontinued, but the people doesn't know what is the metabolic pathway, where when this TBA will be, metabolized. Okay, so and by using the machine learning in 2018, actually the machine learning methods, using the machine learning methods, they made all the metabolic pathways, and that was connected to the linked to the drug similarities. Okay, and they identified that the most probable metabolic pathway for TBI is something like was identified, and there was experimentally proved that the metabolic pathway was TBFA is this it is having two-step process, and in this one step will be done with one enzyme and another step will be with other enzyme, and then he was approved and was published in a Nature, very good article. Okay, and that's how these methods are useful. I mean, in understanding not only the what you can say that how drug is binding and the metabolites, how they are metabolizing. We can predict the metabolism, and then we can see the toxicity. We can early estimate the toxicity of the molecules as such. Okay, and now I just go to that one of the simple, very important drug disease based computational approaches. Okay, where we discuss when you have two diseases, if you are for one drug, okay, some of the molecules which are binding to that, and the other disease, some of the molecules which are binding to that. If any having some commonality, okay, the disease pathway commonality we have, it is sharing a disease commonality pathway. If any new drug which is sharing the commonality of this one or maybe this molecule, then they can bind either of the this one and two. Okay, this is how people will start disease-based commonality thing. Okay, I'll just go through it. Okay, I'll just come with some of the example of it. Okay, COVID in our case study, and then I just wind up top. Okay, I'm a little slow in that. Okay, so simple thing. Okay, this is our IDS repository where we have all collection of databases. Okay, and categorized, and that we are using for different purpose. And one of the based on this, we made a network. Okay, based on our in-house known database, PubChem, Drug Bank, Campbell, etc., and also with the drugs and their metabolites. And we systematically understand the soft targets and their principal targets and their side effects. Okay, what is their side effects? And their side effects leads to any other function and their metabolic pathway. Okay, and then we just systematically categorized them based on their sequence similarity, domain similarity, pathway similar analysis, and protein-protein interactions and text mining. All these things systematically we categorize them. And known molecule database. Okay, and we generated our own SVM model, similarity-based subgraphs, etc. And then we connected to that. Now, if you have a new drug, okay, when you have a drug molecule, when you pass to this particular network, it try to identify a new target. Okay, and if it identify a new target, now we see that which therapeutic area it will bind, and then we do molecular dynamics and docking and do the in vitro and in vivo studies, and then we come up with this is a molecule which is binding. Okay, so one of such molecule which we did it. Okay, we call it CCDS01D3, which was repurposed against the human T cell receptor, a special checkpoint assay method immuno-oncology target, one of the immuno-oncology target as such. Okay, so we use the same network and identify the molecule cancer, and it's not an anti-cancer molecule. It's binding. It is having some other category. Now it is having very good efficacy in the cell lines. Okay, with the IC50 with the nanomolar or something like 8.01. When that target doesn't have any drug, small molecule drug, but it has an antibody, MAP, which is available. And when you compare to the MAP is having an IC50 value, something like 1.569 micro nanomolar. Now our molecule was showing as 8.01 nanomolar. So these molecules are almost comparable with the antibody, and this is a drug molecule. So if you establish efficacy, especially in the phase 2 clinical trial, then this become a blockbuster for the formulation industry as such. This is what we did it in our research for one of the client, and we got it. And another study, just 5 minutes, okay, anti-diabetic drug, okay, glyperamide, okay. We took this molecule and then we compare in a house known database, and we identified a molecule which is very close to that, which is an anti-malarial compound which binds to proline tRNA synthase. And then when you understand the proline tRNA synthase in human, okay, it is mainly for the cell proliferation. It is working on anti-cancer and also other diseases as such, okay, urinary system and cardiovascular, okay. And even with the network analysis, we understand that it is mainly for the cancer. And even it has a VIP domain, especially this transferase has a VIP domain. We find that is there any VIP domain, okay, which target it will bind. Finally, we concluded that proline transferase will targets for the cancer. Now we thought, now which is glyperamide is having very close to the anti-malarial compound, and now we thought that this molecule glyperamide can be an anti-cancer compound, and we did the simulation studies, okay, and we found that this is well binding to the prolyl tRNA transferase, which is a category for the cancers, okay. Now glyperamide actually can be an anti-cancer compound, okay. This actually we did the study, and this is in communication, actually publication, it is in communication, okay. But because I want to show that to the people better, however it is in the communication, that's what I want to show that, okay. This is only for the academic work we did it, that's what I was disclosing here. The previous one is our client work, we could not able to do that. So glyperamide, in the same way, when I see the glipizide is also showing people are using for the anti-cancer, but they don't know on which target it is binding actually, okay. Now we propose that, okay, glyperamide and glipizide, they will bind to proline tRNA transferase and reduce the cancer as such. And some other work we did it, okay, to share the disease with similarity, and also anti-cancer, anti-kinase database, how we connected. But just I will show you a glance for previous one. So this is called as true, polypharmacology. The same molecule, it is binding to different targets. This what we did is, it's called as, we collected all the kinase molecules and clustered based on their chemical topology and also the structural topology of the kinases, okay. Based on that, we did the clustering, and then for the same molecules, how many proteins it is binding, we can get it out. Now, if you put the same drug here in this protocol, then we can understand that this drug can bind to this many targets as such, okay. So it is binding to the PK3, it is binding MK8, the same molecule is binding to the same message, okay, JANK, BLK, okay. So that's how we constructed for our client, and then we got it. In the same way, this just I want to put a glimpse. So domain-based similarity, you know that these are the kinases which share the catalytic site with the hinge region and ATP binding site. So when we analyze that, okay, HIV, especially what you can say, reverse transcriptase, will have a similar domain pattern. When you did the deep analysis, okay, the drugs which are sharing with respect to the etraverin, okay, and also the drug which is for the we can say that anti-cancer molecules here, which binds to the kinases, okay. So they share the same similarity. This BI 4464, now it is a drug, actually is approved now. This and etraverin is sharing the same scaffold. That means, even HIV, from jumping from the anti-cancer to HIV, we can do that here, okay. So that's how we use our computational and these machine learning methods. But I didn't tell much about what are the machine learning. Maybe other people in this, because I saw some of the lectures which will tell more about what is the deep learning methods, etc. And other things, okay. But here I just put it as a application, what we are going to get it and how we need to use it, okay. But I took little longer time, okay, maybe little more, okay, because this is a vast subject, and we need to put it in a smaller way. Okay, sorry for it. And so yeah, questions, okay, right. Yeah, sorry. Yeah, I think, madam, you are in mute. Dr. Suchitra. Yeah, yeah.
Yes, sir. Yes, sir. Thank you so very much, and sorry to obstruct you because you're already 20 minutes ahead from the scheduled time actually. And on the other end, Dr. Girinath sir is waiting actually, and he also joined probably in the starting of the session. And on the other hand, he is listening also.
Sorry, Dr. Girinath, but I have to say the next speech. Sorry. Actually, we were little 10 minutes delay actually. So sorry.
Yes.
No worries, Ravi. No problem.
Hi, Girinath. Actually, after long
Happy
time, I
listening
met
to you.
right,
Absolutely.
right.
Thank you.
Thank you very much.
Thank you,
ma'am.
sir.
Yeah,
thank you, and
right.
kindly stop sharing, sir.
I'll just do that, madam. Yeah, right. Sorry. I'm just leaving the session. Thank you. Right.
Sir, kindly accept this certificate of award and memento from Sun Hills College of Pharmacy for your wonderful talk on AI in drug repurposing and polypharmacology. Thank you so much for joining, sir.
Thank you, ma'am. Right. Bye.
Thank you, sir. Have a good day. Bye-bye. So without a delay, I'm just going ahead with the session of day 2 that is headed by Dr. Girinath Pillai, Director of Gestra Innovations, Bengaluru, and sir is today going to deliver a talk on hands-on session, especially machine learning in drug discovery and drug repositioning. And sir, from Sun Hills College of Pharmacy, we welcome you for this day 2 of ATAL-FDP program, and very good morning to you.
Good morning, madam.
Yeah, very good morning, and thank you, sir. And to introduce Dr. Giri sir to the delegates, I request my dear colleague Shivagami madam to do the honors. And madam, it's over to you.
Thank you, madam. Sucharita madam. Am I audible?
Yeah, okay, madam, you can continue.
Madam, am I audible?
Yes, madam, you can continue.
Yes. A very good morning to one and all present here. Myself, Mrs. B. Shivagami, working as Associate Professor in Seven Hills College of Pharmacy. I feel very delighted and privileged to introduce today's eminent speaker, Dr. Girinath Pillai. Dr. Girinath Pillai is a research scientist at Nairo Research and founder of Gestra Innovations, Bangalore, and formerly a Marie Curie Research Fellow at Malcode Limited, Estonia. Sir carried out his PhD at University of Florida with Professor Katritsky and Carlson, University of Tartu. Sir's work on mosquito repellent with US Department of Agriculture, including SAR and machine learning methodologies with targeted descriptors, attracted global collaborators for scientific findings. Sir was the responsible researcher for 4 industrial projects, Johnson and Eastman in the US. Sir holds 37 research papers in peer-reviewed international journals, 2 US patent, and co-authored a book chapter. Dr. Pillai is expertise in target identification, drug discovery, and drug repurposing by applying a variety of chemometrics and machine learning approaches. Currently, sir, as a PhD student, working under him in the area of aging registered under VIT. Apart from scientific positions, is also a mentor in incubation centers of IIT, Kottayam, VIT, and Government of India's Niti Aayog, ATL schools. Thank you. With this short biography, I would hand over the session to Girinath Pillai, sir. Sir, please take over the session, sir.
Thank you, madam.
Thank you so much, madam. Hope I'm audible and you are able to see my screen.
Yes, perfect, sir. Go ahead.
Great. Just let me hide this. Okay, so before I start, it would be nice if I can understand from which area or which field of interest all of you are from. This could really help me actually to like modulate on what area or what specific details I have to work on. So everybody is from pharmaceutical chemistry, pharmacology, or are there other stream also involved?
So many of the delegates are from pharmaceutical chemistry background, and we have a few participants from pharmacology especially.
Okay, and anyone specifically on bio related? Biology, microbiology? No, none of them. Okay, mostly are on pharmacy. Great. Okay, so I have just went through the yesterday sessions to refer something. So I saw that already Divya, very good known to me, a good friend. She already covered about the target, that is protein-protein interaction and target identification and validation using string stitch channel. So I'm not going to touch base that. And she also showed you Cytoscape, which is a freely available software. And also Surojit covered about data retrieval and chem informatics factors about the chemical molecules. So I thought today I will focus more on to the hands-on, basically to understand how data retrieval is happening and how you're going to correlate with this activity. And also Ravi well explained about GPCRs and some of the repositioning examples also. And I will also show you some extra examples, but still we will a little bit more focused on the hands-on. Today, whatever I'm showing you will be a freely available tools, including paid. You can choose which one to use because you are the best people to decide. Feel free to interrupt me in between. You can stop me. Hey, please tell me what is this? Absolutely fine. Don't wait for the last session. We are already late, but I'll try to finish off before your lunchtime so that we don't want to lose our interest. So without much ado, I'm starting it. So let me tell you, whenever we say AI or all these things in drug discovery, please let me tell you the fact. Don't worry, we are not yet completely there. We are still in the testing phase. We are testing this, testing that. Some work, some doesn't work. If everything is good, we say it worked. So it's like Human Genome Project. It took some time for Human Genome Project to complete, but when it completed, then everything was good. That is what we wanted to understand. The role of scientists is to explore unexplored things as well as to reduce the cost of whatever is being already expensive. That's what has happened with Human Genome Project. After that, sequencing is much, much cheaper, right? Earlier, it's for sequencing, we had to pay up plenty and hefty amounts. So those things have been very materialized. So currently, don't worry, even if you don't know what is AI, what is ML, but if you know statistics, you are the masters. Without knowing statistics, there is no AI or ML. And don't have a imagination that, oh, I don't know Python, so can I do ML? Nothing like that. Do you know to run SPSS or R package or your normal statistical package like GraphPad, all those things? You're good at starting ML. ML means completely statistics, the background foundation. So right now, what we have to concentrate, the biggest concentration for drug discovery, chemist, pharmacologist, or whoever it is, the biggest concentration now required is generating the right data. I didn't say simply data, the right data. Most of the time, the data that we generated is wrong. And I repeat it again, it is wrong. Because we don't repeat the experiments to avoid the uncertainty. Means when you repeat the experiments only, you will come to know standard deviation and error which we have observed from an experiment. Until unless we have a deviation or an error reported, I cannot justify or validate the wet lab experiments. So we need the right data for the AI/ML. Otherwise, the model is not going to work. My slide contains many pictures and others that is taken from different papers, different materials. I don't own any copyrights. It's with the respective authors. The slide is available in my slide share. So you can you will get all the slides, whatever today I'm sharing here in my slideshare. And today I'll be showing you some demo exercises using 9, and that examples are already published in my GitHub also. So these are the resources that you can get directly from my presentation. Everywhere we tell about bridging the gap between the industry and academia. Bridging the gap in the sense it is not giving presentations or training or internship. Bridging the gap means asking the right question of expectations from the industry. What industry needs from academia? What else they wanted to cater? If academia generating a patent, why industry is not buying it from you? It's because you're not answering or solving the problems what industry is facing. Of course, you might be solving the problems of a community, but that might not be always an interest to every industry. So ask the right question to the industry. What do you really want from us? What we can cater for you? And try to solve that questions. And tomorrow your patent makes money. For me personally, and not for me, for many of them, innovation, IP protection, patenting is just for money. And this money is not for us to simply relax, but it's for to re-innovate or invest back in research and development to further innovate. And publication is to showcase our credibility, and we are trying to tell the community what is good, what is negative, what is positive, what are the findings. But coming to patent, you're not disclosing many things. You closed many things, but because for your research, you need funding, right? So there's no wrong in claiming technology license. I agree. And I am also a person who approves and very much forward to open access and open source. But is there someone to fund you for open source and open access? Well and good. So today's main agenda is that to understand why, what, how is AI or machine learning. And the main part is how ML or machine learning is applied in drug chemistry, including repositioning. And then drug discovery, especially to avoid failures, and what to do next with ML. To start with AI/ML, learning denotes that changes in a system, which enables a system to do the same task more efficiently the next time. And this is very important. It's like a kid, right? An infant. They try to walk, they fall down. Next time they try to walk in a different way, again fall down, but they still keep on trying, right? And the same way with us. When for myself, maybe when we were kid, when we started writing, our initially slanting or horizontal lines, everything was not a line actually. It was waves actually. Slowly we started drawing straight lines. So it's on each learning we will try to more efficiently work next. That's exactly what is machine learning is also when compared to statistical methods. So Herbert Simon rightly said with respect to humans as well as with machine learning. Now, why do we need to learn? Because we want to understand, improve efficiency of human learning. And this tries to improve the methods for teaching and tutoring people in the human scenario. But when you're coming to a software or a model, it tries to come up with the data and scientific discovery to uplift or enrich the predictive ability. So when we specify a learning system, we specify the task T, performance P, and experience E. So what we are trying to expect or what it requires is exactly what knowledge is to be learned. Very specific endpoints are required. And then how this knowledge is to be represented, whether this knowledge is represented in activity data or molecular representation data, and how this knowledge is to be learned, which methodology or what kind of approach we have to apply for this to be learned. So that's where even yesterday's speakers, Professor Degani also was concentrating on telling you the different levels of deep learning, machine learning, and artificial intelligence. So I'm not going to cover all these things because most of the speakers covered about this. When you're talking about what is machine learning, it is a branch of computer science which deals with system programming in order to automatically learn and improve with experience. Now, don't worry about the programming part here. I'm sure that most of you will be not much ready to learn programming at this stage. Of course, if you are really good, Now, today, whatever hands-on demo which I'm going to show you, it's 9 based. There is no coding involved, nothing, zero coding. But it's more of a thorough understanding is required. Which input and which output is to be put to what analysis. So robots are programmed to so that they can perform the task based on the data they gather from the sensors, but it's automatically learns programs from data also. And that's the reason challenge that came 2 months back. I don't say which company, they stopped their robotics project just because they got an alarm. Alarm is just this. Two robots started to speak to each other and understanding each other, and they stopped it. And that is a threat. When humans tell something to robots, if they understand and do the task, absolutely fine. But when robots to robots, they talk to each other, this is bad, which we have already seen in the movies like Robot 2.0 and all those things, right? So there was a reason, this is a big news, and they are concerned about it, and they're researching what can be done on that. And that's exactly what I'm going to say at the end of my slide, what is next. Anyway, so moving forward, these are the different algorithms, different approaches that you can apply in machine learning. The main types of techniques in ML, I have seen that already. Divya covered a little bit of explanation yesterday, and she that to explain with examples on supervised, unsupervised learning, semi-supervised learning, reinforcement learning, transduction, and learning to learn. And the algorithms are mainly decision tree, neural network, probabilistic networks, nearest neighbor, and support vector machines. The biggest 3 stages of ML. Now, I don't know how many of you are doing QSAR here. I will reduce my questions. Usually I try to engage with a lot of questions, but due to limit of time, I'm not going to ask questions to you. So the different stages of in QSAR is, the first thing is that model building, get the data and get that right model. After the model is being generated, we have to test them or validate them. And the final thing is applying the model. Major chunk, most of the time, we just publish the QSAR model, but how to apply deployment, never discussed. Very few papers talks about, okay, the reader can put their molecules into our models and then predict the activity. Very, very less. Now, the biggest reason most of the authors say that we don't know programming. We don't know how to make this model available for others to work on it. That is true. That is a bit challenging. So that's where we are missing. So make sure that until unless we apply the model or deploy the model, it doesn't make sense whatever the models that you have generated. So whenever you're publishing a QSAR paper, make sure that your model that you have generated can make the reader to apply it. For example, if you published a QSAR model for anti-melanogenic activity, when a reader reads your paper, there should be an option where they can use their compounds to predict the anti-melanogenic activity using your model. If that is not possible, whatever you published is not reproducible. Yeah, it's difficult. So make sure that the deployment or applying the model is important. Artificial intelligence. Artificial intelligence and role-based inferences is not machine learning. They are both different. So do not confuse between machine learning and artificial intelligence. Now, let's look into the machine learning in drug discovery. So there was always a question whether machine learning is overhyped. But let me tell you at this time, because this is back in 2018. Yeah. But now I don't say machine learning is overhyped, but AI is overhyped, very much overhyped. Everybody now, even whoever not worked on AI also started putting, I'm an AI enthusiast. Now, when I was doing QSAR studies back in 2012 and 2010, we were using support vector machine, neural network and all. That time we never said that, oh, we use machine learning, but we rather was proud saying that we use statistical methods, right? But now, even we use statistical method, we say that, no, I'm using machine learning. So that's the difference. So hyping is there, but make sure that we do it the right hype. Now, this is a survey that is being taken from the Chemist and C&EN journal magazine. Now, I don't know whether there are participants who have already listened to this earlier, this slide deck. but there's a small difference in the coming few slides on the drug repurposing. Now, majorly more than 40 to 43% people actually accepted and agreed that it is being overhyped, right? And few others said they did not agree to that. So still it is the same, that some agree and some disagree. There are many drug discovery companies worldwide, and many of them concentrate and focus on specific opportunities. For example, like us, Nairo Research India, then Immunocure, like Ravi's company, we all worked on focusing on drug discovery. Some on software side, some on consultancy side, some on coming up with the novel molecules. All it's a mix. But ultimately, to look at the cost saving analysis for all these companies is that how to implement new technologies. Artificial intelligence, where the combination of deep learning and machine learning, it's actually going to give a good implementation for a long-term savings. And cloud-based technology platforms is also going to save us some in the infrastructure. Where Ravi was already mentioning about Google Cloud TPUs, which is a way ahead than what we talk about GPU. So the cloud-based platforms are also going to help us to save time. But this is something very alarming in the distribution of CAD services worldwide. You can see target validation and target identification. We don't have many players in the market. We have very few players, right? But for target identification, lead optimization, we have many players in the market. So that's a big difference that we have to focus on. And this is where deep learning algorithms being applied on molecules in different stages. If you see here, you take the molecule, you can have molecular design, material design, then drug design. And also you can look at accelerated quantum mechanical calculations, conformational exploration, QSPR. You can also do reaction prediction, reaction optimization, retrosynthesis. I'll be discussing about that in a moment. At the bottom of the slide, you can see it's a normal standard QSPR. You have the molecule, you have the experimental activity value, and you apply machine learning models to correlate among these properties of the molecule and the activity, and then you correlate it. So it's like a training cycle between predict, evaluate, and optimize. And then we have to go for a schematic representation of the main components of atomistic machine learning. So unless we know how to interpret these pictorial representations of the clusters, as well as correlation plots, Williams plot, we cannot interpret what we are trying to translate here, because interpretation is something very important when you're coming to drug discovery based machine learning approaches. So make sure that we know what we are trying to say or convey. And keep in mind, most of the time when you do QSAR also, okay, QSAR or SAR models, we end up with one model and we predict or discuss only about that model in the paper. Make sure that you try to build at least 4, 5, or 6 different types of methods being applied, and all these models were being generated. That is very important. Don't think that a single method is going to suffice for all kinds of data or methods. So make sure that when you're starting or doing a machine learning model, different techniques or different approaches to be applied on the same data, and which one is more favorable, reproducible, that has to be taken. It is not that, yeah, my software that I purchased is only offering this method, so I'll only use that. Nobody said that that is the best one, and nobody going to accept that is the best one until unless you tried all the method and publish and said this this is the best because of 1, 2, 3 reasons. So make sure that you have multiple models or multiple methods being tried in machine learning, not only in drug discovery, any method, any data. So then you have to choose the right method. Now, what is the impact of AI in CAD? Even though I said AI is not there yet, but still people are in the verge of investments and trying to work on it. AI identifies drug candidate in few weeks, around 46 days, they were able to identify a lead candidate. Now, it's in the kind of a preclinical or clinical trials. And exploration of the chemical space, basically, what we say is, if you want to download a Zinc database or a binding DB database, it's like 6 GB if you download that file. And that contains might be, maybe 90 million compounds or 9 million compounds. But when you say chemical space, The file size of the chemical space will be just 30 MB, 30 MB, but it can generate 17 billion compounds. So the difference here is the building blocks are there in the chemical space. The algorithms can generate all the combinations, possible combination, which is having a 80 to 85% of synthetic feasibility. That means if you predict 100 compounds using this chemical space AI, you will be able to synthesize at least 80 of them. So that is a success rate, and it's tremendously successful. 80s and all is absolutely fine with this approach. So up to this, do you have any questions before I proceed to this particular, I'm sorry. Yeah, before I proceed to any of these drug repurposing facts. Just a second. Yeah. Is there a, okay. Oh, okay. So there are pharmaceutics, biotech, and everyone. Okay, great. I will watch this chat in between. But if you have any questions, please unmute yourself, or you can put the text in the chat. If you didn't understand something, please let me know. I don't want to cover all the slides, but I want to make sure that whatever I'm covering reaches you. That's all I want. So moving forward, let me hide this. Okay. So drug repurposing, why we are doing drug repurposing or repositioning? We want things to be fast and inexpensive. When you're trying to compare between the new drug development and the repurposing, you can see the biggest difference. First of all, success rate. That itself is the best reason for repurposing. For the new drug development, when you have 10,000 compounds or the candidates, you end up with one molecule success rate. But for repurposing, when you have 10 drug candidate repurposed, at least one success rate. So 1:10, and there it is 1:10,000. For a new drug development, it's unknown safety until unless you go through clinical trial. We don't know the safety. And of course, we would have done toxicity studies on animals, in vitro, cell lines, blah, blah, blah. But on humans, that's what matters for the drug development. So unknown safety there. But in the repurposing, we already know safety because it is already being in the market. Pharmacokinetics, dynamics, toxicity, efficacy, it's all well known, being it's already in the market. And the years to impact is 1 to 3 years only for repurposed drugs. But when coming to new drug development, it is somewhere between 12 to 19. Even though we say implementation of AI, we can get a drug into the market 4 to 7 years. But let's wait and see how the success rate comes in. All the proposed dates are proposed term duration. It's all interesting. Now the cost, it's much, much cheaper for repurposing when compared to the new drug development. So this is the reason why everyone is looking at drug repurposing. And that is not so easy. And even Ravi also mentioned that there are a lot of bottlenecks, and we have to have checkpoints to check which one is better, which one is bad. And we need to have a thorough strategy to identify the false positives and the false negatives. Moving forward, very, very interesting paper. Computational and practical aspects of drug repositioning. So this is published back in 2015, and which tells really good about the computational approaches. Some of them already covered by Ravi. So we need to have a better understanding about structure-based, pharmacophore-based, as well as network-based analysis. But when you're coming to drug repurposing, there's a different levels. There's a classification. Classification of drug repurposing claims according to the scientific evidence. This is called drug repositioning evidence level. Based on this only, we can say how far your compound that is repurposed is acceptable. I would recommend, if anyone is working on drug repurposing or repositioning, please make sure you mention this DREL level in your paper. That really attracts to the industry because they completely look at these levels. Now, if the DREL level is 0, that means there's no evidence, but includes some in silico prediction without confirmation. Let me tell you, 60 to 70% of our publications are on that level, 0, because we have only in silico studies, but no wet lab confirmations. Now, when coming to entering to one, which some of them I have already done, mostly on in vitro studies with limited value of predicting in vivo or human situations. But mostly it is in vitro, in vivo is very minimal. Then when it goes to level 2, animal studies with hypothetical relevance in man. And number 3, incomplete studies in man at the appropriate dosage. That means prove it's a POC stage. Very few cases or inferences from medical records, some clinical effects also being observed. For example, 2DG, they did it some way, something similar to that. Number 3, it is still incomplete because they don't have the complete study that is required. And of course, it is also being repurposed because back in 1979, it was looked at as an anti-cancer drug, and they found that it is toxic at an above a certain level. So they stopped that studies, not in India, outside of India. And later in India, some of the teams, they looked at this for repurposing, and then they tried to work on it. Anyway, so moving forward to the level 4, it is well documented clinical endpoints observed for the repurposed drug at doses with safety limits. So if you ask me, the best level to start with is number 2. If you have number 2, DRA level, you're ready to talk to an pharma industry or industrialist to talk about the next level to make it 3 and 4. But until unless you have number 2 level, it's going to be very difficult for you to propose this for a POC, that is a proof of concept. So make sure that we do it in the right way, and that's where we try to get the best done. The drug, now what is the repurposing actually? So the repurposing is basically to find a new clinical use for the approved drug is what we look at the repurposing. And also, what is a drug rescue? So repurposing and drug rescue in similar, what we are trying to see is that many of the drugs fail at efficacy level, right? And sometimes we know that they are safe, but efficacy hits bad. So in that cases, there are 2 levels of drugs that is being done. One is for diagnostic level, and the other is for therapeutic level. So some drugs which has been failed in the efficacy, some still being proposed for diagnostic level. But I know some groups in ICMR in India, they work extensively on drug rescue purpose. So I don't know whether you're aware of this terminology, drug rescue, which is trying to find a clinical use of a stalled clinical development stage compound in phase 2 or beyond, established PK and tolerability, maybe safety, and also usually a known chemical structure. So I would also propose a new domain where chemists, medicinal chemists and pharmacologists can work together, including with the other clinical experts, where they can look at to rescue the drugs. And that's a great innovation because you are going to make some modifications. You have to do synthesis, you have to test it and prove that it's better than the one which is already being failed in the phase 2 or beyond. So a drug rescue is also another area that we can look at. And when you're looking at the target based on specific on the target, if you're on target, we are trying to finding new use of a drug acting through the originally known target based on literature, omics experiments, or for if it is literature, of course, you can use string, stitch, disgene, and databases. Even Divya and Ravi was showing you some networks with the targets. So those yesterday, even Divya also showed you about the string databases, stitch databases. And so these are all freely available online. And the positive feature is that it is likely to be compatible with dosing of the original drug. So that is on target. And in looking off target, we are finding new uses of a drug acting through a novel or unanticipated drug target. It can be based on docking and fingerprints. Ravi was showing you how he was able to identify the similar molecules, which is again fingerprint-based methods. And drug was not originally optimized for that target, but so need to be careful of dosing. So ultimately, whatever we are doing computational, sorry to use the word, it will be junk until unless we validate it using in vitro or in vivo studies. Computational predictions are just predictions. They might be good, might be bad, but validated. So being working on the computational area for more than 16 years, I have to admit this, that it is good if you do it in the right way. It is really, really, really bad if you are not considering the right data and not doing it in the right way. So don't think that softwares will take care of your mistakes to be corrected. No, until unless you give the right data and until unless you interpret it properly, software also can make mistakes. Software is just a piece of tool to do calculations. That's it. Interpretation is our perspectives. Now let's see what is this drug repurposing process looks like. So we have, let's imagine we have virtual or physical library of drugs, manually curated or from Campbell, and then we try to do a virtual screening, or without doing virtual screening, since we already know they are approved drugs, you can directly go for biomolecular screening also. And then you try to understand what are the possible new target activity. But now the biggest challenge here, the question is, you have some drugs. Now we need to know what are the possible other drug targets that I can look forward to the biomolecular screen. So in order to identify the neighboring or interacting other proteins apart from the dedicated protein target, you can use these online tools called as String and Stitch, where you can try to form a network and then try to understand the transcription factors, hop genes, so that this will be responsible or interacting partners for this drug molecule. Maybe similar drugs. Sometimes if it is not there in the database, then similar ones also can be looked up. Later, we go for confirmation of clinical effects. Then it goes to phase 2B, phase 3 clinical trials, and then finally it comes out as a repurposed drugs. So this is the process. Now, let me tell you, most of the time we are stuck with the first stage. That is, we have some physical or virtual library of drugs. We do virtual screening. Boom, done. Where is a biomolecular screen? We might be doing a cell-based assay, but that is nowhere going to correlate your virtual screening with a cell-based assay. It gives a little bit of control, but not completely. You still need to do a target binding-based studies. Now you might say it's expensive. Of course it is expensive. Of course it will be time-taking process. And that's how the drug repurposing works out. So make sure that you have the right data that is being published or put forward to patent or proposal to industry. That makes it more interesting. And also here I have to tell you an example. For example, bimatoprost. Bimatoprost is a compound that is being used in Lumigan, which is an eye drops that is especially used for the reduction of elevated intraocular pressure in patients with open angle glaucoma or ocular hypertension. So this particular drug, which is eye drops, is especially used for that one, for ocular hypertension or the open angle glaucoma. But they were able to later repurpose it for some interesting application. And that interesting application is that the same compound was at a different concentration, were able to use to treat hypotrichosis of the eyelashes, to make the eyelashes to grow, including the length, thickness, and darkness. So something for the beauty aspects, right? So rather than artificial eyelashes, people who are not having less thickened and all, they use the same molecule, but with a different commercial name. And this is repurposed. This is the best example that I can find it in internet. Of course, we have many others, but this is already in the market, right? So earlier it was used for treating ocular hypertension, but the same when it is applied on the eyelashes, it is increasing the length, thickness, and darkness. Okay, now you need not to find lattice. I think it's not available in India. Now, what is the pharma requirements of a drug? So we first and foremost thing is that we want to understand the delivery route, whether it is oral or parenteral, topical, or inhaled. So topical is basically skin, on skin. Then parenteral means intravenous, those are parenteral. So these are the delivery routes that we are looking at. Then we also want to understand drug-drug interactions. Then differentiations from current therapies like safety, cost, efficacy. Then dosage frequency, whether it is 8 hours, whether it is every day or weekly or monthly. What is the cost of the goods? Risk of misuse of controlled substances, because aspirin or ibuprofen, as the dosage increases with respect to your body mass, then it's going to be risk actually. So tolerable side effects, whether it is safety in pregnant and breastfeeding populations, dose adjustments in pediatric patients, and monitoring, especially on specialist use, liver function test, etc. So when a requirement for a drug is this much list, of course, the list is further big, but very, very perspectively, I have given this. Now, when you go for a repurposed, the same thing applies there, right? So we have to see how these all can be controlled, not only in silico. Now, let us see how machine learning is applied in drug repurposing. So already Ravi showed you some examples on doing docking, chemical similarity studies, target identification, as well as exploration studies, including pharmacophore. But here, let's look at more into a machine learning approach. First step is data preparation, because when I was one of the coordinator for drug discovery hackathon by MHRD. So during that time, the biggest challenge by most of the participants for the machine learning and AI projects, they were saying that we are not able to get the right data. Getting data is very difficult because we have to read papers, get the data from paper, manually curate it, and then do the model, which is very tough job for us. For a computational science or a computer science people, it is difficult job, because for them, curation of data is something a time-taking process, and they need a good team for that. So they were expressing their difficulty. That's why I was starting my slides with that, saying that generate the right data so that they can use it. Anyway, the next one is building a knowledgeable graph using different lenses to see the data clearly. So whatever data came in, we have to categorize it. We have to curate it. We have to clean it, and we have to automate it. And then we also want to see whether AI ready data, because simply bringing in all data doesn't mean that it is ready to do machine learning models. No, we have to clean it. We have to look at missing data. We have to see whether there is unrealistic data. We have to make the program to understand whether it is a continuous data or a categorical data, a nominal or a real number, or an integer or a float. All these things has to be defined. Then finally, you have to create an AI model tool which runs a machine learning to make a trained artificial neural network. Need not to be artificial neural network. You can always work on CNN and many other methods. For this is an example that I've taken from a paper, so they have used artificial network. Then finally, we are putting a reliable prediction in the hands of the experts. So using this model, we are trying to predict what could be the possible target, and then try to identify that. That's exactly what we are trying to go to see now. In the first step, we are trying to, for example, we are trying to correlate the data from different parts, and then, like Pathway Studio, Reaxis, PharmaPendium, Open Targets, to try to collect the data. Not necessarily you have to collect the data in this way. There are many other resources too. And then the second method, to build a biological graph. We want to relate between compound, protein, disease, and adverse event. So Cytoscape can be very well used for this. Yesterday, Divya was showing you. So you can get the data from multiple resources, and you can merge the network to come up with a sensible graph to look at the data clearly, how the compounds are related to specific diseases and the proteins. And the third one, whether we have the right data for machine feeding. So we want to have a strategy that can we predict new drivers for the disease, can we predict off-target interactions of a drug that are genetic drivers of the disease, or can we predict if a drug treats a disease via any drug target or through any mechanisms. So we want to have a clear picture about the strategy, and then we look at how we can generate the model. So we are trying to understand the fingerprints or descriptors of these each drugs, and then we try to find the new interactions with the new targets, because we are doing a repurposing, right? And then we try to take the target descriptors from the amino acids, and then we try to match each other to understand the new interactions. And finally, we come up with a similar repurposed targets. For example, lacosamide. Lacosamide actually inhibits SCN3A gene, also inhibits CRMP, but it also predicted binds to CFTR as well as CTSV, and it is related or regulates the chronic pancreatitis. So the disease is there on the green color, and the middle region or center one, which is completely on the target genes, and on the left hand side you have the molecule. So this gives you an idea. These are the possible targets that could be repurposed, right? It's not that simply, okay, I'll take a antifungal compound and I will do anti-cancer. We need to have a thorough understanding. Why are we doing the antifungal drug to an anti-cancer? So we need to have a backup plan or backup data to review and understand how this is being correlated, or how did we end up deciding that antifungal drug molecule can be also acted as an anti-cancer drug. So before moving forward, any questions? I don't see any questions. Are you able to understand? If you're able to understand, you can put number 1 in your chat. If you are not able to understand, if you want me to repeat something in a very legible way, you can put number 2, so that I will try to cover this, whatever is being missing. So please be open. Don't worry. The session is for you. So make use of it. Yeah, I can see very few people saying 1. So the others, then it is number 2 then. So I think I have to repeat it. Okay, slowly coming in. Thank you so much. Yes, I can see it too. Thank you so much, being very open. Absolutely, we require that. Okay, if you can tell me which part or which region is that is confusing to you, I'll try to cover that up. Of course, not taking too much of time. Please be open. So others also will be helpful. No? Okay. You just said 2, but I think you're typing, so please go ahead. I'll come back to this chat and look at a bit later. So if you want me to look at something, step 4, okay. Thank you so much. So step 4 is creating a tool for running the machine learning to make it trained. Okay. So I didn't explain that in detail. That is true. Let me tell you this. So after generating the model, we have to deploy it. To be example, in Python, we use something called as pickle, and then joblib, and then we try to put it on a Streamlit or a Heroku to deploy it. And once we deploy, we get a certain data from the user who is going to use this model. And that is what we call it as a web app. So when I say here as an AI tool, it is a web app reflecting, making sure that it will try to take some input data. The input data will be the features. Features means what all the compound or what are the existing targets, those details. And then it will be able to put it into the model, and it will come and predict and tell you, okay, these are the possible other targets that you can focus on for your molecules. Now, this is already available in the database. What we know is publicly available, that is string and stitch. Please, I am asking you, in YouTube, you have yesterday's session. Please go to Divya's session. She quickly told about how to retrieve data from string and put it into Cytoscape. Even in my YouTube channel, we have discussed in detail about target identification and target validation. So I would recommend you to go through that. But what the 4th step is actually deployment. So whatever the model that you have generated, you're deploying it as a web app. Of course, you need to know some amount of coding there for customization. Okay, great. Moving forward. Now, how do we transform, or how do we make machine learning algorithms to understand, hey, this is a chemical compound, there is this information, how do we make them understand? Only through 2 ways. Either you have to convert your chemical structure into numerical data, that is called molecular descriptors, or you can convert them to fingerprints. Now, whatever I'm showing here for that structure on the top, where the easy to understand by the CPU, it's a fingerprint, right? They're general, compact, unique, invariant, smooth, and fast. But when coming to chemical compound, we all know that each regions are very specific. Some have keto groups, some have carboxylic acid groups, some have hydroxyl groups. So each one is unique. So we want to have a unique representations for each of these regions. So then we will have sub fingerprints to represent, and then a global fingerprint for the compound. Then only we can be very selective when we are going for doing structure activity relationship. Any questions? No? Okay. Someone tried to unmute. So moving forward with the drug discovery, how to avoid the failures. Now, why we are doing all this analysis? But in all this analysis, you understood that we need data. Until unless we have existing data, we cannot do any additional wonders. So all things are worked on existing data. As I told you, what is learning? From the existing data, when we try to learn better and we try to do it efficiently next time, we are trying to enrich or improve whatever we were trying to do. The same thing, we need existing data, otherwise we cannot do. Now the question comes here is that as a civilian, when they go to pharma store or drug store, they wanted to understand the two things: whether this drug is going to save my life, is there any side effects, and is this drug is affordable to my pocket, or is it going to be very expensive? How can I reduce the cost of this medicines? Now you will say AI, you will say in silico methods, you will say computational chemistry, etc., will come. But these are all methods which is already existing. But still, are we able to reduce the cost? No. The biggest reason that we need to understand is from the existing knowledge. When you read your papers like a literature review for your new research work, the same way we need to also understand how the drugs are being successful and how the drugs failed and because of what reason. When you find and go behind the reason, then you'll have a clarity where you have to make corrections. Example, the cause of failure. Yeah, the cause of failure for oral drugs. Early between 1960s and 1980s, most of the rejections happened just because of poor pharmacokinetics. Luckily, after that, we have all these QSAR models came in on a local QSAR and mainly 2D QSAR, not 3D. Please keep in mind, most of the very well validated QSAR models are 2D, not 3D. 3D is good when ComFA came completely on a pharmacophore-based screening analysis, not the way we do the 3D, that is load the smiles, convert 2D to 3D, then generate descriptors. No, not the right way for biological activity studies. Just think about it, because any compound binds to a biological target will form a bioactive conformation. So you have to get the bioactive conformations descriptor, not a simply converted 2D to 3D descriptor. Anyway, that is something that we have to out of the box have to think about it. So make sure you ask why in each step so that you will be able to getting the justifiable answer. So the first and foremost challenge we had was pharmacokinetics and ADME, and also we had issues with the efficacy. Later, back between 2000 to 2010, the major reason of failure was because of toxicity and strategy. So when we were able to implement structure activity relationships using statistical methods, we were able to come up with a lot of QSAR models for predicting ADME and pharmacokinetic models. But then we came up with an issue called toxicity. Now for toxicity also, we do have QSAR, but not that accurate. Then came with the knowledge basis, for example, Direct Nexus. Of course, Direct Nexus is a paid software through Stardrop, but they are knowledge-based. Knowledge means from the clinical trial, they come to understand what are the different 43 human toxicity endpoints, and based on that, they try to predict. So that's where we have different drug discovery approaches. I'm not covering this today because that is not our theme. And also when you talk about drug repositioning, when we say that lock and key model, we always know one lock will have only one key or its copies, that's it. But one lock cannot, or one key cannot go into multiple locks. In 1894, this particular definition for molecular docking was coined. But now in 2020-21, we cannot accept this, because for the same one key, we are trying to put in two different locks. Already existing one lock, which we know, The other one we are trying to repurpose or reposition. So it's only half the truth. So we have to advance our understanding and perspectives as the technology is being looking forward to. So I'll just skip this because these are just included, but I want to cover the next few slides, which I'm going to cover in the basically on the hands-on. Just a second. Madam, can you tell me how much time we have? It's 11:50.
Sir, it is up to 1 we are having a slot time, sir. Hello?
Hello?
Yes, sir. I am Sunil Kumar. Up to 1 we are having a slot time, sir.
Can you hear me? Hello?
Sir, is I audible, sir? Hello? Sir, I can hear you, sir. Hello? Sir, Girnaz, sir? Hello?
Am I? Okay. Thank you. Okay,
Yes, sir.
fine. So I was not hearing from us. So I think it's up to 1 o'clock.
Yes, sir. It's
So
up to
I will
1 o'clock,
quickly
sir.
get into the demo session also. So if you want to know what are the success stories of the CAD, for example, we say a lot about computer-aided design, but then is there any drug in the clinical trial or others which we can say it as an example? Yes, there are plenty of them. More than 112 molecules which has gone to the clinical trial. Even the computer-aided drug design helped them. Now, don't think that the computers or softwares help them to choose a single molecule to go to clinical trial. No. Computers selected maybe 10 or 15 or 20 or 30 molecules. Among that 30, this one has gone to the clinical trial. So this I'm telling for the very layman people who are just starting with this area, others who are already aware of this. So it is very important that when you look at different stages of computer-aided design, you have multiple tools required. Don't think that with single, for example, just using Autodoc or just using StarDrop, you can get a drug in the market. No, it depends. So we need multiple stages, multiple tools, multiple approaches to come to a conclusion of 30 or 20 or 40 molecules. And later from that wet lab, in vivo, in vitro studies, you come to get the final set of molecules for clinical trials. So these are some of the examples that I can tell you that has entered into clinical trials based on that. Now let's go through some of the hands-on experience with NIME. So NIME is a freely available package. Anyone can use NIME. It's called analytics platform, free for academics too, and even for researchers and others. And I am one of the partners for NIME in India, especially on the life science and drug discovery. So I'm happy also to share about this. If you have any other doubts or if you need any teaching materials or some course materials, you can always reach out to me. Now, when you're looking at what are the specific drug discovery nodes, nodes means you can say a package, for example, or a tool is the following. So you have 3D ECAM, Alva Science, then CDK, Analose, Earlwood, Indigo, LASA, NGS, RDKit, and Bernalis. Now, all these are freely available nodes. The paid ones or the commercial ones are Chemaxon, Biosolve-IT, then Cresset, Molsoft, Schrodinger, and MOU. These are all paid nodes. So unless you have paid license from them, those will not work. The others, whatever I have listed here, yes, they do work. Right. So now, what are the functionality it is? So it also, we have to see how these particular models can be applied and how these all can be referred to it. So it's very important to understand like what are the different functionalities of these different nodes or NIME nodes. Vernalis can do kind of data retrieval and understanding the conformations or generate conformations of the compound. And when you go for Earlwood, you can do structure activity relationship analysis, including Indigo, where you can generate descriptors and do similarity studies And RDKit, you can again generate descriptors, you can clean molecules, also optimize geometry. CDK also you have Ambit as well as many other molecular property generation tools. And we have some basic chemistry nodes where you can draw the structures using ChemSketch or Marvin Sketch and Structure Sketcher. Then there is also other reading format like SDF file format, MOL2, MOL file, Smiles, and many other formats. There are also converters available from molecule to CDK. OpenBabel is also available in KNIME, where you can convert from SDF to mole, multiple moles to single SDF. All those things can be done. Mole converters, and you have also viewers, like if you want to view 2D structure, 3D structure, protein ligand interactions, pharmacophore, all those things can be also looked in KNIME. Right, so these each ones are important for us to not the analysis tools, but these are all input and output nodes. Now there are chemistry calculators. As I told you, there are descriptor calculators from Pero, Enilos, Vernalis, and many others. Because as I told you, we want to convert the structural information to numerical data. For that, the machine learning models will understand that. If it is not able to understand those molecular properties or features, then we cannot correlate them. And then we can always also predict some of the values here. So based on US EPA, Environmental Protection Agency, you have some of the developmental toxicity models and some few predictive toxicology models are also available, like carcinogenicity, skin sensitization, fish acute, and fathead minnow, and others. Then the output port, for example, if you want to write after all your analysis, you want to write them as an Excel sheet file, or you want to write them as a mol2 mol file complex or SDF, all this can be done, and which will be telling you which are the data types. So I told you in machine learning, we need to generate the data and we have to define what are the different data types it is there. For example, whether it is a string data type, whether it is an integer, or whether it is a float, whether it is a categorical or classifying data, all those things has to be defined properly. Now, if you ask me, what is this? This means it's a workflow. Workflow means each boxes are called as node. That node performs a task. When we connect that node with another node, whatever the task the first node completed, that will be transferred to the next node as input for the next node to perform the task. So it is a flow. That's why it is called a workflow. Each node performs each task, and each one has its own properties or tasks to perform, and then it passes on the data to the next node. That's how we get connected here. So here, if you see, it started reading from mol file or SDF file. I did not connect. We can connect any one of them based on the data that you have. And then we try to connect it to RDKit from molecule. So it is trying to convert from a mol file to RDKit format. Then we are trying to add hydrogens in the next node. For example, this node, we are trying to add the hydrogens. And then we are trying to do a 3D geometry optimization. After doing that, either we can view the structure, we can write it as SDF, or it can go to an interactive table and we can write it as a CSV file also. So at a single click of a button, I will be able to load the molecules, optimize it. I can write it as CSV, including the energy values, SDF file, and view it. If I want to do something else, so I can convert it to 3D, from 2D structures to 3D, I can convert. Then I can make CDK to SDF, and this is a different way of optimization. So two of them tries to do optimization, but two different methods are being used. Similarly, if you have a SDF file and we can convert it to canonical smiles, and these canonical smiles can be used to predict certain models activity. Now, whatever I'm telling is not machine learning. What I'm telling is how to generate the data or how to load the data. And many a times, data manipulation is very important. That is called as data transformation, data wrangling. So this is what I was trying to tell you, how to prepare your data, which can talk to your machine learning models. Not all the data, simply putting in Excel sheet doesn't make sense that it easily being converted to a machine learning understandable data. So we need to do either transformation, wrangling, missing data. So all those things has to be done. So there are certain nodes like joiner, group, ungroup, then splitter, partitioner, aggregator. These are all different nodes available within 9, which can help you do data manipulation. Now, if you want to generate different conformations of your compound, the same can be done. You can draw the structure, you can convert, you can do the conformations, and you can filter the conformers based upon the conformation energy. If you want to also predict the site of metabolism, similar ways of nodes are available like SmartSIP and WITSIP. And if you want to do a similarity search or substructure search, also can be done within the 9. If you want to do a pharmacophore screening alignment, can be done within 9. If you want to calculate different molecular properties, join together and then save them as a single file to do your QSAR modeling, that can be also done within 9. So these are all very straightforward aspects. Now there's another tool called as Orange. So in 9, the advantage is that you can generate data. Okay, you can also do machine learning. You can also use Veka. I know most of you will be aware of this program if you have been working on machine learning models earlier. Veka is a very old package, still very active in the user community. Then R package is also implemented into the 9. So this is a combination, but most of the thing just now I showed you is that generating data in 9. Now, how can we apply this generated data to machine learning? For that, I used Orange. Orange cannot generate data of chemical compounds, but it can do machine learning models and deployment can be done. The same thing can be done in 9 also, but I thought this will be very easy so that you have different 2 softwares which you can combine. Here we can do feature ranking. Very, very important step. It's sometimes called as feature selection, or it is called as descriptor selection. So features in machine learning, let's imagine you have descriptor calculated from your molecular structure. All those descriptors are called as features. Then you have corresponding experimental activity data for these compounds. Those experimental bioactivity data is called as target, right? So in other ways, what we say is in statistics, all these features are called as X variable Y variable, and all the the bioactivity data is called as X response variable. But here the response variable is a target, and the descriptors are the features. Now we might be having predicting for 4,000, 3,300 descriptors, which is called as features. We don't want to use all of them. We need to know which is the appropriate one that can correlate with my activity data. So feature selection or feature ranking is something very important. It is all depending upon imputation also, because we want to see the missing data, right? As I told you, drug discovery, the biggest challenge is we are having very sparse data. So based on the sparse data, sometimes we have to impute, or we have to see how to balance the missing data also. So this can be also done within this particular package. Of course, it's free. Then we want to do a classification tree, which combines interface of visualization of classification tree with scatter plot. This can be done. And then you can do a cross-validation. Cross-validation means you will try to do multiple methods, and then you are going to do a test and score to see which one is having the better predictive ability based upon the cross-validation. And then if someone is very much interested in Python coding, of course, you can do Anaconda also. Now, before we go to the demo, I also want to emphasize on, especially on this. This is all which you know, AI implemented here, virtual reality, augmented reality, protein structure solving, fold, protein fold, etc., etc. But this is something that is very much concerning. This is not my personal view. This is completely based upon a consortium where OECD and where they came up with this particular future forecast. What they're trying to say is that by 2000 or after 2040 or 50, they are always expecting humanity at risk. And this is exactly correlating with the example that I told you. The robots started talking to each other and understanding each other. So this is something that everybody looks at, but good thing is that European Union has came up with a AI policy, artificial intelligence policy. There are many things that is being reflecting what is being shown in this slide. So I'm sure every country will be coming up with these policies. Like in India, we have startup policy. Then we have genetic engineering policy in European Union. Similarly, gene editing or therapeutics policies like that. I'm sure AI policies will come and have a balance on technology and human sustenance. Those, by saying this, I want to conclude my presentation saying that we have to consider the quality initially with a weightage of 25% and more of a diversity of 75%. And as the size of the lead reduces, you can consider to give more weightage to score and less weightage to the diversity. Consider the enrichment factors, very important, because enrichment factor is not only in docking. Any kind of virtual screening with the decoys that you choose or subset that you choose, we need to understand the enrichment factors. Good synergetics between human expertise and computational tools are important. Don't think that computational tools can itself do wonders. No, unless you put in your information, it cannot. Avoid missed opportunities. Don't put unnecessary methods, unnecessary filters. Since other papers used, I will also use the same thing. No need to. Sometimes it doesn't work. I don't know how many of you know that Lipinski rule of 5 can be used for looking at oral bioavailability, but not simply filtering your database to docking for docking. So we have to use the right filters for the right time, for the right opportunities. Otherwise, we will be missing the compounds which might be required for our analysis. Understand the significance of parameters or properties. Whenever I said the feature selection or feature ranking, most of the time they do statistically. For example, which descriptor is more correlating to your bioactivity. But keep in mind, not always a correlation only works. That is where every time in every sessions I used to tell causation versus correlation. To give a very funny example is that correlation means you can correlate the number of oranges grown in Canada in this month versus number of babies born in this month in Canada. Both can be correlated. But is there any logic behind it? Is there any meaning behind that correlation? Nothing, zero. So that's exactly what we're trying to do in drug discovery also. Make sure we have both correlation as well as we have causation. So when you're correlating and saying, I got an R square of 82, good. But then make sure that you also have a reason why these features are correlating to the bioactivity. If that features does not have a physical meaning or a physical significance to your activity data, then you're missing the causation. So in drug discovery, whenever you're doing machine learning, make sure there is significance of the properties or features that you're selecting for your model. So don't only look at the correlation, but also look at the causation. It's very, very important. And that's the reason why most of the QSM model are predicting wrong molecules that they are active. And that is what we call it as false positives and false negatives. False positives are still okay, but false negatives are dangerous. So make sure you have causation or significance, understanding or correlation in a logical way between the descriptors or features with respect to the target or activity, then your models will be more meaningful. Evaluate and decide the tool or the approach. As I told you earlier, there are different algorithms, different methods available. For example, random forest, decision tree, support vector machine, artificial neural network, Gaussian process regression method, AdaBoost, etc., etc. Which one is the best? Yeah, I cannot simply say random forest is always the best, or I will blindly say a decision tree is the best because it always gives you best R square. But let me tell you, it's not good model actually most of the time, because R square we always get 99, 90, 95, but I don't personally prefer. Not for all data, but for some data it is fine. So we have to evaluate all these models and which one is giving you better prediction, better reproducibility, better accuracy, less error. That's what we try to take it up, right? So make sure that you try to understand all the possible tools or models. Now, if you say I don't have time, then we are doing research. You should find time. Then check the reliability of the data used. Whenever you're using a data, It might be from papers. It might be from databases. Some humans are working on it. There might be errors coming. Even when I was working on QSAR modeling, I have found many errors, and I used to write to authors. Some respond, some did never respond. And interestingly, one of the major paper that I was about to put everything and about to publish the data. Then I realized that, and they didn't mention that. I did realize that they took a log of logarithmic value. For example, they took the data from another paper. That paper already had a log value. Okay. And then these guys took it and put another log value on it. I don't know why. But then it is like multiple logs, and it's a completely wrong data. And then I was initially starting with QSAR, I'm not aware of many things. So then I put the data and I came across with the models and all. Later, when the residual plot came in, we were trying to able to identify that, no, this data is having submission. Then we went to the original back reference and then tried to get the data. We saw that there is a mistake in the published data. So we have to be very clear the reliability of data used. Otherwise, we will be repeating the same mistake others have done. So that's all from me for the presentation part. But now I will go to a demo to quickly show you how to generate the descriptors, how the generated descriptors can be correlated with the activity data, and how you will be able to generate the machine learning models to understand how this can be predicted and others. There is no coding involved, no Python used. It's all plug and play nodes and others. Now you need not to install this right now. You can later, whoever is interested, you can install, and this is freely available on the internet. So do you have any questions before I move forward? Okay, I don't see any questions. So let me share my software. Can you see the screen? I hope you can. Now, I don't see any chat. Are you able to follow? So I'll be explaining this one by one. And this is already published in my GitHub. So it's already available in the public domain. And this is the software called KNIME, K-N-I-M-E. Yeah, this is software called KNIME. And I'm using 4.3.4. The latest version is 4.4. You can go to this website called knime.com, and then you can fill up the form, and then you can download the software directly. It works for Windows, Linux, and Mac. The latest versions works only on 64-bit, not on 32-bit. And if you know Python, it makes wonders. If you know R programming language, better. But if you don't know both of them, still it works. So let me quickly show you. This is a flowchart that I have done for retrieving smiles of the Campbell IDs that you give. Okay, I'll explain in detail a bit later. But before that, what are these nodes, from where it is coming? Let me explain that. So this software has different parts and all those things. This middle region where I'm scrolling now, this is called as a workflow space editor, where you can put the nodes, connect each other, make text and explanations, all those things can be done. So similarly, you can have multiple workflows that can be also loaded. Okay. And now if I click on a specific node, you can see the explanation of the node on the right-hand side. So you will have the quick help menu. So make a practice when you do this, you read this at least about what is the input required, or what could be the possible output that could come out. It's very important for everything that looks nice, of course, but reading is also involved. So if I do, this is a scatter plot I'm able to generate from a machine learning model that I have done. But R square is only 0.49. That's okay, because the descriptors are very limitedly used. I'll explain about this workflow a bit later. Now, at the bottom of this, you have something called console, which tells you what is it working on, what is the error, and then is a node monitor. For this particular node, when it runs, it will tell you what it is going on. And if some calculations going on, we'll get to know the progress. And outline is like a navigator. you will be able to navigate between the different spaces of workspace. On your left-hand side, something very, very important. At the bottom, you have something called as node repository. And this is where you have chemistry nodes, you have community nodes like this. Now, when you install KNIME, you won't be finding any one of them here. So when you're installing, after installing, you have to download and install these nodes. For that, you have to go to Help menu, click on Install New Software. A new pop-up window will come. Make sure you're connected to internet and click here to say all available sites. It will search the internet and then it will be able to tell you what all the packages that can be downloaded to this particular version. Now, please do not install everything. It will unnecessarily take a lot of space in your hard disk. So try to install only the ones that you require. No need to put everything now. Now, I cannot explain each one of them what it does. It takes too much of time and we have a time shortage. So I have to consider that. So I will quickly tell you, you have to click on each of these workflow nodes and then or the partners, and then you will be able to understand what each does. As I told you, some are paid, so no need to unnecessarily download the paid ones unless you have a proper license from the respective vendors. Okay, I think my internet is slow. Oh, loaded. Great. So here you can see we have BioSolve IT nodes. Of course, I have license for that, like CSAR, docking, confirmation analysis. Then you have KNIME extensions like Amazon. Then you have chemistry nodes available, like you can see here. Then you have deep learning, including Kera, TensorFlow. And yeah, if someone is working on bioinformatics, then you have some on NBI, NGS. In chem informatics, that's where this is all I have. I have installed all of these nodes. Vernalis is actually having an update. That's why it is highlighting. And then we have image processing. If someone is working on images, cell profiler, deep learning, all those things are available. Then few other community extensions are also available again on Java, again RapidMiner, then R packages, and other lab extensions are also GitHub, Python, or Jupyter Notebook, even Pipeline Pilot. Yesterday, Divya was showing you Pipeline Pilot in her slides. So Pipeline Pilot can be also connected here. Even someone is using a dashboard creation like Power BI, that can be also done. So many integrations are available. Even Molport, I hope you know Molport database, right? Where you can purchase compounds. Their nodes are also available. If you're using Padel for descriptor calculation, that is also available. So these all can be clicked and then next, it goes and initially installs it. Once you get installed, you restart, you will find all these nodes listed out here. So this is how the basic installation process. Make sure you have Java installed before installing KNIME. Okay, so let me see if any questions. Oh, thank you so much. If you're understanding, I'm so happy for that. Great. So now I hope this is clear with the installation and the other part of the software. Now, you know how to start. You can go to File, click New, and then you can always start with a new KNIME workflow. Next, you have to give a name. I'll say FDP demo. Make sure you don't give any space, either underscore or without space. You can give the name and then click on finish. Once you click on finish, the empty workspace editor is there. This is what you see here. Now you need to know what to do. Now, if you want to read an Excel file, I'll just type Excel here. I can see your Excel reader. So I can drag and drop here. Drag and drop. Or I just double click. It does, yeah, in Windows it might work, but not here, not working. Then let's say I wanted to read a SDF file. I'll just type SDF and I can see here SDF reader here, right? SDF reader. I can bring in SDF reader, right? Now, if I want to do a descriptor calculation, so I will say descriptor. Sometimes it is descriptor, sometimes it is not descriptor. Okay. So here I have RD-KIT descriptor calculation. Again, I'm dragging and bringing it here. So when I clicked here, I can see on the right hand side, it is giving me some explanations. I drag down and it is saying me the input port should have the table that is containing RD-KIT molecule. So SDF file format is different from RD-KIT molecule, right? So that's what basic understanding of those things are required. So whatever on your left hand side is always input, whatever is on your right hand side is output port. So we want a RD-KIT converter here because it requires RD-KIT molecule. So what I'll do is I'll go to community nodes. I will drag down and take RD-KIT, and here I have something called as converters. And the converter I have RD-KIT from molecule. That's what exactly I will have to do. Once at the end, if I want to convert RD-KIT format to CD molecule, I can take RD-KIT to molecule. So now in order to convert my existing SDF file, I'll take this and put it here. And I will connect this to this one and RD-KIT file to the descriptor calculation one. I connected all this, how to arrange and all. It's all how you look at it should look better. Now I have to give input. So what it does, this will read your SDF file from your computer. It will convert your SDF files to RD-KIT format, and then it will try to calculate all the descriptors. Now, after having these descriptors, I wanted to write it as a file. I can either write it as an SDF writer, so that it will again write with the structure information and descriptor information. Both all will go here. Or if I want to write it as a CSV file, because sometimes I want to take it to some machine learning programs. So I want to write it as a CSV writer, both ways. And then I can put it here. Now, so these two I will come a bit later. First, I have to load a SDF reader. So I'll double click on it so that the configuration came. It is asking which SDF file you want to read. I already have some sample files here. I'll just load this one. So make sure it is SDF file. It can be 3D, it can be 2D, all those things you can look at. Use a molecular name as row ID. If you are given a name in your SDF, then you can use that. But let us see what data we have. When I'm going to property, I will say extract all properties and it will try to scan. Of course, I don't have any additional property other than the structures. If your SDF file, whether you want to use what kind of coding characters, like by default it is UTF-8, or it depends upon which particular software you used. And then you click OK. Once you click OK, you can see the traffic signals, red, green, yellow, then it becomes green. Red means it requires data, it to be configured. Yellow means it has the data and it is also configured, ready to run. Once it completes a run, then it becomes green color. That's the process. So now this is yellow color. I click this, click the right button. I can say execute, or click the button, click the node and say on the top, you have an execute button here, just execute. Now it tells you it is green color. Green color means it has read the SDF file. Now click the right button and click at the bottom, you can see red molecules. When you click on it, it will show you the possible molecules that is being read from that particular SDF file. Right? Let me see this. Yes, you have the two, because these structures, some are 3D, some are 2D. So I have the structures. Those are, you see that. If I want to save this data as an Excel sheet, of course I can do that. Now I will again go to RD-KIT. I'll double click so that I will be able to configure. Now it is asking which column contains the molecular information. So I automatically only molecule. If you want to put a new name, I will say RD-KIT mode. So that tells me that that is a RD-KIT converted mode from the SDF file. And then I can ask to remove the source file so that this will be helpful for us. So I will say, okay, remove the source so that there won't be two different structures in a single file. And then in the advanced, if you want to say whether to keep hydrogens, yes, I want to keep hydrogens. Whether you want to also look at aromaticity and stereochemistry to be corrected, you can do that. I click OK. So it is yellow color. I click this and it is run and it is completed. Now I want to do a descriptor calculation. I can double click on it. What all descriptors you want to calculate? These are something called as group descriptors. I am not going to consider that because that has challenges when you try to save them as Excel sheet. I don't say you need not to use them. You can use them. After that, you have to ungroup it. These are all grouped descriptors. So you can then, using a node called ungroup, you can ungroup this. Otherwise, it will not save it as CSV file. So when I mouse over it, you can see these many descriptors are embedded within that particular single descriptor. So I'm again using RDKit mode, which is that is convert one, and that's it. Now I'll execute it. The descriptors are calculated. Now I'll click the right button and say molecules with calculated descriptors. Now you see the structures with all the descriptors calculated. These are all descriptor calculated from RDKit, right? So this is how the descriptor calculation is being done. This is a small data set. That's why it was so super easy to do that. Then now I want to write this, the structures with the descriptors to a SDF file. Double click the SDF file and you have to say where you want to save. So I will say, I want to save in demo FTP. And I can say whether to use the same thing, whether you want to use a column as molecular title, whether you want to omit properties. I want to omit properties with missing values. I don't want them. And if there is no structure, then I wanted to say there is no structure also to identify that. And I can say which all properties to be written to the file or not. I can click OK. Similarly, I can go to CSV file. I can double click and say where or which CSV file to be written. So let me say I will write it here. And demo. Also, I can expect and limiters given. Advanced, whether you want to quote it in strings, of course, you have to quote the strings. If it is string value, if it is text value, make sure you quote in double quotes. And that's all. And then click OK. So here you can click and play. It was saved. It is saved as a SDF file. And this one also you can click and save. This will be also an SDF file. Sorry, CSV file. So both these files are now written, and then you can use it for other softwares and make use of it. Now, this is how we will be generating the data, saving, loading, and calculating the descriptors. Any doubts in this area, like whatever we did right now? You can unmute yourself. I think you participants have the rights to unmute.
Yes, sir, they can unmute, sir. Dear delegates,
Hello.
if you're having any doubts, please unmute and ask the queries. Thank you.
Hope we are all connected.
Or else the delegates can post in chat box also.
Yeah. So no doubts. Okay, good. So I don't know how many of you are attentive, but I don't know how many of you are already knowing this. If you're understanding, I'm happy. Great. So this is about loading the data now. Now, how do we apply this data? Now, this is a different workflow with the same concept of the previous one, but now we started applying some models. So what I did is I started getting the data, smiles, I converted to SDF, RDKit from molecule. Then I started calculating the properties of descriptors. Then I combined all the different properties, looking at duplicates and others. Now, this is the portion where I want to explain about the machine learning part. So I wanted to partition it. So this is a node for cross-validation loop. This cross-validation loop is very important because of what I was telling you, the machine learning methods try to self-learn by themselves. Whichever the model is getting you the better accuracy, then it stops. Until unless then it will see all permutations and combinations of features. So for that we use this X partitioner. So when you double click on the X partitioner, you will be able to see how much number of validations or folds of validations to be done. And there are three different methods: linear sampling, random sampling, and stratified sampling. Okay, here I'm giving only 10. This itself will take some time. And then I'm trying to do random forest for the first. I'm not going to do everything. I'm just doing a random forest. Here, as I told you earlier, I wanted to do the modeling on my Ki values. I have a Ki values in nanomolar, which is taken from Campbell. So I'll be choosing my target as log Ki, and I have to choose which are the descriptors I have to choose as my feature. Not necessarily you have to choose these many descriptors. You can reduce that. Just for the sake of explaining and showing you examples, I'm doing this. If you want to remove some descriptors from your model or that not to be considered, you can select this and you can click this button so it gets removed, so it won't go to the model, right? And then I'm not applying because it's already being run because it takes time. I have already pre-run it to show the results too. Then, so first we will try to learn machine learning, right? Learn. And then we use a learned model to predict. So when you go, this is a regression base. So here we just tell which column it has to generate. What is the name of the column to predict the value based on the forest model that we have generated. And then we put it as aggregator. Aggregator is telling you what is your target actual experimental value and what is a predicted value. And then we bring it to a scatter plot to see how they are correlated. Now, when I run this, it will be running multiple iterations because in the X partitioner, I told 10 times. So it will try to look at 10-fold validations. So when you click a single button, it tries to do automatically 10 iterations. To each time, your R squares will be different if you stop in between. So this self-learning mechanism is very important for cross-validation. When I'm going to show you orange package, there also we have this cross-validation mechanisms. Let me put it for load. It will take 2 minutes to load. Right. So after this all information, so we can go to this aggregator, click the right button and say prediction table. So this is a table which tells you there are chemical structures which is taken from Campbell. I have its actual nanomolar value. This is the actual nanomolar value. And this is the log of the actual value. Others, all our descriptors are used 9 to generate the descriptor. These many descriptors were being calculated. And I can see at the end that this is the predicted value. And this predicted is the log Ki value. It's a predicted log Ki value, not the real Ki. Okay, that was faster than expected. Okay, let it load. Okay. This I'll explain in a moment. By the time, let me finish the 9.1. So, okay. Yeah. So, so we have the predicted value. Now, we wanted to see what is the quality of the model. We can view this scatter plot by saying that, okay, this is my x-axis will be the actual log value and my y-axis will be the predicted value. And you can see here the R square is 0.68. Not that great, but still acceptable, better than nothing, right? Something is better than nothing. But we can still optimize because I can see there are a lot of outliers in this model. We can always look at it. See, we already know that removing outliers will improve the R square, but it shouldn't destroy your chemical space. So when you're removing an outlier, make sure that you have a valid scientific reason why this particular compound is being removed as an outlier. If you're not able to justify that, your outliers are just to improve the R square, but not logically meaningful. So it's a tricky game. That's a sad part here. That's why I tell all my students also, QSR is one of the dangerous method. You might get addicted because of the R squares or statistical parameters, but many more things are involved into it to look at its reliability and reproducibility. So this is all about 9 to give a quick overview. But again, if you want to validate QSAR models, then again we have, so here I have used a linear regression method, right? And rather than random forest, and then a regression predictor, then I splitted them. Then I looked at certain criteria. So these are certain criteria which tells you applicability domain of your model based upon Alexander Tropshaw's paper. Then similarity, looking at again applicability domain, understanding the similarity among the compounds within the dataset used in the QSAR, and model acceptability criteria. Now, in my case, my R square is greater than 0.6, so the model is acceptable. R square CV external should be greater than 0.5, and all these differences should be less than 0.1. And then Ki and K dash value should be between 0.85 and 1.15. So if you are able to satisfy all this criteria, then your model is acceptable. I don't know whether I ran this. No, I have not set the criteria properly. No, yeah, and that data is not yet prepared. So based on this, we can automatically make a NIME to check whether our QSAR models are meeting this criteria. Unless it meets, we can go on partitioning it so that the validation, cross-validation happens repeatedly. Similarly, many more methods can be applied. So let me show you what are the other machine learning methods that we have. So where it is, analytics, statistics. So these are all statistical methods what we have. Then under mining, yeah, this is where you have a ocean of different methodology. You have Bayes, naive Bayes, you have clustering method, you have rule induction, which is fuzzy rules, you have neural network methods, you have decision tree, then ensemble learning, which is PMML, and then linear description, linear polynomial, logistic regression, principal component analysis, support vector machines, and many more. And then let me show you Veka also, just a second. Yeah, so in the integration, we have Veka. Veka is again an offline open source tool available for machine learning algorithms. We have classification algorithms. Again, Bayesian logistics, we have naive Bayes. Then Gaussian process regression, one of my favorite method for QSAR, Gaussian process regression methods. Then you have other fuzzy methods and metadata methods, classification algorithms, AdaBoost, and many other decision tree tables, clustering algorithms, association, predictors, many more. And apart from this, we have something called as water, H2O, and I didn't install it, I think, where we have AdaBoost and others. AdaBoost is always there, okay, already there in Veka also, so we need not to look at. Then Plotly is a graphing tool, which generates very much interactive plots and graphs, which can be, which is based upon Java as well as Python. So this can be also put into the 9. So it's a big ocean. We cannot finish within 30-40 minutes. I can only give you an overview. Of course, if you go to my YouTube channel, you will find 3-4 more videos explaining a little more detail about 9. Now, coming to Orange. Orange is a package where you can do machine learning much more easily, and not too much of what to say, coding, but it does not generate data. For example, if you have used a PADL, if you have already generated some data, that data can be brought in here as an Excel sheet or CSV file or TSV file, and then you can generate the model. So that process can be done here using Orange. Orange is also freely available package. Here you can do most of the machine learning as well as statistical package. I've been using these tools back since 2008 and 2010 extensively, early during my PhDs and other studies and for some of the works. So that time it was very minimal functionality. It doesn't look like this, but now much improved and a lot of functionality features being integrated into it. A lot of automation. This is called as automation actually. Every industry requires automation. So what all kinds of models that you can generate, like constant CN to row algorithm, calibrated trainer, KNN, Again, you have decision tree, random forest, gradient boost, SVM, linear regression, logistic regression, A bias, ADA boost, neural network, stochastic gradient, trackings, and you can save and load the model. So these are the different machine learning techniques or approaches we have inbuilt within Orange. The good thing here is, of course, it also is there in 9. I don't deny that it does not have, it does have, but for a new person, it will be a little bit learning curve is there. But here, the deployment of the model is so easy. That's why I wanted to show this. I'll start from there and then go back to explaining what is the different things here. The similar context of 9. You have different nodes for different tasks. They are connected each other by connectors. So what I'm trying to say here is, using ADA boost, whatever model is being generated, I am able to save it as a model, save it as a file. And later, I will load the same file, which is a model. I can share it with anybody. They can load this as a model. And I will be just inputting the descriptors of my new molecules. What is the purpose of QSR? For example, we have experimental bioactivity data for some of the molecules. Using that, we generate a QSR model using the training set. That is called training set. And this model is generated to predict activity for other molecules, which is yet to be synthesized or yet to be tested. So we are just seeing whether the activity would be good or bad based upon QSR. That's the purpose of QSR model. So until unless I apply this model somewhere to predict it, I won't be able to understand which compound I have to choose. So that is what we call it as deployment. So this portion, this region is called deployment. We deployed the model and we are using this model using the descriptor, because descriptors are required in order to substitute into the model to predict the values. We just gave the descriptors, that's it, because the structural information is through descriptors. We don't have activity value for this particular set of files. Using the model, using the descriptors, the algorithm or the package is able to predict the activity for the new set of molecules you have given. And that is where you are applying the model. Until unless you apply the model, it is going to be very challenging, or the big question mark, why did you generate a QSR model, right? If someone cannot use your model, then what is the purpose of publishing a QSR model? Otherwise, you yourself would have applied or used for QSR model. So to go back to my paper back in 2014 or 13, which was published in Chemistry, a European journal. So there we predicted relative abundance for cyclization. We came up with a model. There we used some existing data, we generated the model, and we predicted the relative abundance for some of the structures which we are planning to synthesize. And based on the QSR model, we chose only 2 or 3 molecules to synthesize, in which we had a pipeline of around 14 molecules to synthesize. There were predictions were bad, so we went for only 3 based upon the QSR prediction. And that came out to be good. That just because we used only 2 descriptors, just 2 descriptors with good correlation. Not necessarily everywhere we should have only 2 descriptors, right? So that journal is having an impact factor that time around 7 or 8, I think. I don't remember now how much it is. So it was widely accepted, and I can see many people are still using that model to predict the relative abundance for cyclization. That is exactly what I'm trying to tell here. So just predicting the model doesn't, or generating the model, it has to be applied also. Now let's look into how this works out. You can go to data and you can drag and drop the file as like what we do in 9. I am just deleting it because I already have a file here. I can double click it. It opens up the file as you can see on the screen. You can load a CSV file. I have some examples already have worked on. So you can have a CSV file. And then this is the important factor, which as I told you, you have to prepare your data accordingly what it tries to understand. We have to say which is your endpoint or which is your target. You will have only one target in a data set, right? So you have to define what is your target. And you have to define which are your features. Features means which are your descriptors, right? And then you also can tell, okay, I don't want this descriptor, or this particular acid I don't want. And this is a name. I don't want to be considered this as a descriptor. I'll say skip, or I can say metadata. Metadata means it is representing something. So I can skip also if I want, or I can define it as metadata. So this preparation is very important, not only here, even in NIME or in any machine learning packages. Your data preparation is something that you have to spend a lot of time. Once you have done this, and then it automatically gets applied, or I will click on apply, so it gets applied. I think it's going full screen. Yeah. And then if I want to view the data, of course, I can go here and view the data. Double click it, I will be able to view the data. So I can view the data. What is the endpoint, and what is the log p values, and what is the range, everything I can view it here. Now, I have come to select columns. Here, this is again very important. Even though I know what is target, what is my features, within the features, I can think of removing some data. Here, I have the red ones are colored in numerical data. The green ones are colored as categorical data. Now, even though in some of the descriptors, some are zeros and ones, they are categorical data, yes or no, right? Or active or inactive, present or not present. Number of hydrogens is not categorical because it says 1, 2, 3, but there are multiple. Still, the software tries to understand as a categorical data. So you can change the data type also, whether it is categorical or continuous. So here I have selected some of the data set just for an example. Okay, so you can choose which column, which data set. If you want to see a comparative analysis, you can generate a heat map, and the heat map you can say your rows to be based upon what clustering, or it should be split by what type of methods, or what type of colors you want to look at. All those things you can define. I'm not going deeper into it because it takes more time for analysis. You can also do principal component analysis. How much components you want to consider based on the number of components you're choosing. It will try to tell you the clustering analysis. And then you can also take a radial plot. You can plot between which different properties you want to take, or you can define a different shape, or based upon different properties. You can also define based upon the size and label. All those things can be done here for visualization. Now coming to this particular thing, basically what kind of methods we want to apply. We want to apply AdaBoost, neural network, support vector machine, random forest, and decision tree. If you double click on this, you will have only very few parameters here, because I hope whoever working on AdaBoost knows about number of estimators, learning rate, and which algorithm to be used, whether it is linear, square, or exponential. So before giving those parameters, I would recommend you to please go through each of these methodologies, optimal property that you have to give. Of course, we cannot cover this all at this time because it's already 1 o'clock. So I don't want to cover this each and everything. It's going to be very difficult. So you can see which could be the optimal one. Then we are trying to do a cross validation here. After each one of these models are being predicted, we go to test and score. So here I can say how much number of fold. I said only 3, but I can improve it to 10, 20, 100, but it is going to take a lot of time. But based on this, I'm able to predict the R square. The highest R square came for neural network. I don't believe because too much of descriptors there. AdaBoost is something acceptable. A random forest is pretty much acceptable if everything comes into the picture. So 0.88 is something coming for a random forest. Okay, now let's see some visually, because something is predicted. Now let's look at the scatter plot. Now this in the scatter plot, I can choose whether it is based upon AdaBoost is best or not. And in my y-axis, I have my endpoint. The endpoint is the actual experimental value. Now I will try to take random forest. So random forest, you can see my R square, R is equal to 0.96, but when combining both test and training set, it is coming to around only 0.88. I can color them based upon log P or shape, size, everything can be. So your pictorial representation could be made dynamic, where you can put a lot more information into your scatter plot to explain, okay, this region of the compounds are having more log P, and this one is having less log P, and the shape based upon your blood-brain barrier, if you want to mention, wherever it is negative, it is dash, and wherever it is positive, it is plus. All those things can be defined. After that, each models we want to see what is a predictor. You can put all these things into the single prediction tool. It will be able to predict each one of them. After that, based on which one is performing well, for example, if you're trying to choose random forest is the best, you have to connect the random forest with the save model so that the random forest model will be saved, load the same model, and whatever descriptors are coming into the random forest model, you have to predict the descriptors for your new molecules, that's the purpose, right? And then when you combine these two for the prediction, it will be able to predict the activity or the target value for those set of molecules that you're going to predict. So this is how you generate. And this workflow, what I'm showing here, as well as what I've shown in the 9, both are available in the GitHub. Now let's look at finally, let's for 10 more minutes. Finally, let us look at a ready product, a tool. Whatever I showed you is something happening in the back end, or we have to look at a POC level. POC means proof of concept, or we are doing a researching and all. When it comes to a production level means it came to a final tool, right? It came as a click and drop and tools. So that is where we have something called a star drop, where all these models are generated already. Again, you can also do a QSA based modeling where it uses random forest and many other machine learning methods to predict, including toxicity. And it also generates a chemical space, which also explains about the mechanisms, rule-based inferences, and also like a neighborhood joining method kind of machine learning methods. They try to combine to see how the structures are coming up. So this is where a software which can predict ADME properties, human toxicity endpoints. You can do multi-parameter optimization. You can also check which compound is the structure activity, because I told you earlier, right? Each region is specific to a specific fingerprints. So it will be able to understand which region of the compound is more selective to toxicity or more selective to solubility. That can be identified. And you can also do a pharmacophore screening on a 3D, and you can also predict regio selectivity of a compound, whether we of a pro-drug strategy, whether the stability of the molecule, which atom is more stable and less stable. And we can also design new molecules based upon the property. Again, exploring the chemical space, exploring the reactive database, as well as reaction libraries. By defining the reactants, it will be come up with a product, but this product is completely based upon a property. For example, we are saying that we want to design a new or a transform a molecule or a lead optimization of a molecule, specifically considering ADME and toxicity properties together, so that we need to have a balanced properties for the newly proposed compound. So the algorithm and the machine learning based methods will be able to generate a new compound which has a balanced property of ADME as well as toxicity. Once that is generated, you can always go back and do docking and see the activity studies, and then finally prove that out. And here also auto modeler. So here also we can predict the generate the models, and later the generated model can be applied here so that it will be listed out as a model that to be applied. So within the software, there is a generation of model, and within this software, there is a deployment of the model and applying the model. 3 are also, this is as a package, it has come as a finished product. So all these are possibilities. Now it is our choice which one to use, how to take up the predictive ability. So that's all from me for today. But if you have any more questions, others, I'm happy to take it up. Thank you so much.
Very wonderful, sir. Thank you so very much. And
I think I'm not able to hear you. Let me check.
Can you hear me, sir?
Sorry, I'm okay. Is someone speaking? I don't hear. Hello?
Hello, sir. Can you hear me?
Okay, I'm not able to hear. Just a second. Okay. Can you hear me now?
Yeah, we can hear
Okay.
you, sir.
Now I
From,
can hear you. So far, I
yeah,
was not able to hear any one of you. Sorry.
thank you, sir. Again, so very wonderful talk, and people are actually requesting for a good hands-on session. I think we met with an objective of all the delegates, and yesterday one of the delegate requested me that if possible, give some idea on Gromax actually, sir. So for a 5 or 10 minutes, can you put some idea on
Ah,
Gromax?
okay. Okay.
Specifically, they asked, so just requesting you.
I'll do that. No problem. Are you able to see my screen?
Yes, sir. Perfect.
Okay. So I will give you the references where you have to look at these things because it takes more time to explain it at this moment. But let me give you the result. I'll post the links in the chat also so that you will get much more detail. So first thing is you have to go to my GitHub MD notebooks. Now, let me give you a little bit marketing here. So, what Madam is trying to say here is that when you do not have a good computer, I mean, everybody has a good computer, but for Gromax, enough resources for a computer, and if you do not know how to run Linux or how to install Gromax, but still you are very enthusiastic to learn molecular dynamics, I found an alternate way where you can run MD simulations on Google Colab. Now, Gromax also has cited this particular Colab, which I'm going to share with you. So you can use that as a very good learning tool. So you have to go to this link, and then you can see here there is a link called Open in Colab. So I'm just posting this link in the chat first. So you have to go to this link for Google Colab, which works with Gromax. Use it for learning purpose. You can use it for your research work also. Once you learn the limitations of Colab, Colab has a limitation like every 12 hours, the data gets reset. If you're not active in front of the machine, it gets killed automatically. It's free, no? So if something comes for free, we have to take a pay. It's not paid. So when it is free, you have to take all these challenges, but it does a very great job with the performance. It can do 154 nanoseconds per day on lysozyme. So that was a performance. You need in order to do any single scripting here, all scripts are already into this notebook. You have to just execute it and change the PDB ID and define which is the force field that you require. But otherwise, all the files, all the result analysis, including graphs, result analysis, everything is incorporated. I am working on principal component analysis also. I'll be incorporating that too in the same link. Nothing is going to change. And including 3D visualization, everything is available. So similarly, I have also released for protein ligand simulation using NAMD. Just a minute, let me show you that link also. So if you go to NAMD in the GitHub, that is just loading. Shouldn't take this much time. Okay, so here you have a Colab NAMD, and then I'm copying and pasting this in your chat window. This is for NAMD. This is also similar to Gromax. Here you can do what is that is protein and ligand simulation. So all the supporting files are here. So if you go back to this, you have something called binding free energy calculations. If you go to binding free energy calculation, all the configuration files are given here. You need to just prepare the input and you need to just run this configuration file. And there are 8 different calculations to calculate the actual binding energy binding free energy of the ligand and the protein complex. Now, in the NAMD, the only challenge is that most of the input file you have to prepare offline using VMD. Now, how to do and all, I'll show you. So using VMD software, which works in Windows. The good thing with NAMD is that it works for Windows, Linux, Mac. In GROMACS, it will work only for Linux and Mac, but not in Windows. In Windows, it works through Citrine. Very workarounds are there, but I would recommend only Linux on that. But NAMD works for Windows also. So the challenge here with NAMD is that once you prepare all the files, you have to load it to your Google Drive. All the instructions are given here. After putting it to the Google Drive, you will see the files like this as I have shown it here. All these files will be uploaded to Google Drive, and these files are required for the Google Colab to run the calculation. Now, there are 4 different calculations goes ahead, because these 4 different calculations starts with first one is minimization, second is heating, third is equilibration, and the fourth one is production. Unless you finish the production with NVT, that is ensemble with velocity and temperature, you cannot consider that for your protein ligand calculations, right? So all these initial 4 calculations has to be performed, and that can be done using Google Colab. Now, you might ask me another question, how to prepare the input files, how to do that. For that, you have to go to my YouTube channel, where I am posting the link again in the Zoom chat. And there you go to playlist. If you go to playlist, there is something called as Molecular Dynamics GROMACS and Molecular Dynamics NAMD. If you go to Molecular Dynamics GROMACS, you will get the explanation of the Google Colab using GROMACS. If you go to Molecular Dynamics NAMD, you will have 3 different videos which explains about the different steps involved in preparing the input files using VMD. All those are explained in 3 different videos. And I was telling you that binding energy, free energy calculation has to be calculated. And you can see each one runs for 50 nanoseconds, 100 nanoseconds, 200 nanoseconds, and it takes too much of time. So this can be compromised or run in Google Colab for you. But production run, it's a bit challenging. As I told you, if you leave the page inactive, it just dies. So it might not work properly. So there are challenges. Of course, something comes for free. There are difficulties there. So these are the resources that I wanted to refer. Since ma'am mentioned, so NAMD and GROMACS can be used even on your mobile. If you're using an Android phone, if you're using Chrome browser, this link works there also. So from your phone, you can run a molecular dynamics simulation, at least for learning purpose. That is always good. So that's all from me, madam. I'm still not able to hear. I don't know whether you're speaking. Okay.
Yeah, sir, can you hear me?
Yes, I can.
Thank you so much for addressing my request, actually. And thank you so much once again, sir, because this is the 3rd time I think you're hosting all the delegates and giving a wonderful session again. Probably I will take much more from you in future also. There is a lot more to hear from you.
Everybody is learning every day, and we have to repeatedly hear to get it registered in our mind. So absolutely good. Thank you.
Wonderful, sir. Wonderful. I think participants, if you have any queries, you can just unmute your mic and you can straight away ask to sir, or you can also post in the chat box. Sir, I have one question. Sir, may I know what is the website to download Orange?
Let me post it here. Because when you search for Orange, it will be different links will come. That is confusing. Let me give you the correct website. Yep, it's there in your chat window now, the link to download Orange.
Yeah. And delegates, any other queries? We're running short of time of 2 minutes ahead actually to the schedule. And sir, can you please share the details like your mail ID so that delegates can contact you for further queries if you are interested.
You can write to me this email, and you can WhatsApp to me to this number. When you're sending some error, please send me the picture of the error. Otherwise, I cannot understand what could be the error. If you simply say it is not working, I should know where it is not working, and you have to tell me which step it did not work. Then only I will be able to help you. And please excuse me because you might find a delay. I might reply, but I'll reply as soon as possible. Thank you.
Thank you, sir. Thank you so much. And really, it's an honor to have you on this ATAL-FDP platform. I hope all the delegates have a wonderful session of day 2. And from the management and principal side, I just taking this opportunity of presenting the certificate of award and a memento to use it. Probably this might be a less, but still, it's an honor for us. Kindly accept it.
Absolutely. Thank you. Thank you so much. Happy to be here.
Thank you, sir. Thank you so much. Have a good day, and kindly assist to Central College of Pharmacy in future days also in the form of webinars or FDP programs. Want to hear from you more, much more.
Yeah.
Thank you so much.
Thank you so much, madam.
Thank you so
much. Bye,
sir.
everyone. Take care.
Have a great day, sir. Bye, bye. Bye, bye. Bye, bye, sir. Dear delegates, it's up to the morning session of day 2, and I request all the delegates to join for the third session of day 3 by 2 p.m. And you can use the same Zoom link for afternoon session also. And a few delegates are requesting me to post the feedback. The feedback will be posted after the completion of the afternoon session in the WhatsApp. You can find the feedback link in the WhatsApp. And I hope there is no queries from the delegates. And yeah, see you all in the afternoon session. Bye-bye.